This is a production of Cornell University.
Okay, good morning, everyone, and thank you, Maura, for that introduction. I'm very excited to talk to you all today about some of the work that my lab is doing and turning to tomatoes' genetic past to improve traits for its future. So let's see. Before we get started, I just want to thank the grad students for the opportunity to present here today. It's really an honor. When I was a grad student, these symposia were a highlight of my graduate career. This is the first one that I attended in 2016. The theme that year was breeding for the emerging global middle class. That year, I had the opportunity to nominate and ultimately invite a scientist whose work I really deeply admired. That's Frank Mangan at the University of Massachusetts Amherst, who devoted his career to reaching out to immigrant populations in the Northeast United States, developing market opportunities for new vegetable crops for those communities. Um, I learned recently that Frank passed away last year. This symposium gave me the chance to meet him in person, hear a wonderful seminar. So, to be on the other side of that and presenting today, it's really meaningful to me. So thank you all for the opportunity. And thank you, Corteva, for sponsoring these wonderful programs. So I was also excited as a new tomato breeder to hear the theme of this year's symposium. Maybe I'm biased because of what I work on, but I think tomato is really a model for the conservation, characterization, and utilization of wild relatives in plant breeding. Um, Thanks to the efforts of pioneering geneticists like Charlie Rick, who collected thousands of accessions, both wild and domesticated, of tomato. We have access to excellent germplasm resources in gene banks. Wild relatives of tomato have been used to answer fundamental questions about evolutionary biology, like the evolution of self-incompatibility and interspecific incompatibility. And breeders have used these genetic resources for, at this point, almost 100 years to identify traits of agronomic importance in wild relatives and bring them into domesticated tomato. So just to give a really high-level outline of my talk today, um, I want to share a little bit about the genetic history of tomato before going into kind of what the modern U.S. tomato industry looks like and where the research that my lab is doing, where we aim to kind of fit in there. I'm going to talk about two different projects that we're working on. One looking at a very diverged tomato wild relative and looking at sources of abiotic stress tolerance there. And then also going more recently into tomatoes' evolutionary past and breeding modern varieties for quality and beauty using heirloom genetics. And then finally, I'll talk about some of the challenges that we're facing and what I think possibilities are for the future. So they're generally considered to be 16 species that are wild relatives of tomato. These are found in Western South America, where populations of these grow from the western slopes of the Andes to the coast. Two of these species are also found exclusively on the Galapagos Islands. These species occupy a variety of different ecological niches and grow in a variety of different microclimates that people have characterized extensively, considering what that could mean in terms of their potential impact for tomato breeding. So in terms of the domestication of tomato, for a long time, it's been recognized that the most closely related wild relative is Solanum pimpinellifolium, which you can see on the left here. This species produces small, maybe pea-shaped fruit that ripen to a red color. But there's also been this other botanical variety of tomato called Solanum lycopersicum variety cerasiforme that grows either as a cultivated plant through South America, Central America, Mexico, or it grows in wild populations or as like a weedy species in relation to human habitats. And it's been proposed that this botanical variety was kind of an evolutionary intermediate in the domestication of tomato from Solanum pimpinellifolium, but the exact nature of where and how that happened has been under debate for a little bit. One of the most recent theories that's come out proposes a pretty complicated story of how tomato was domesticated. But the story that they present is most parsimonious with the genetic data that we have. So what the authors of this paper propose is that tens of thousands of years ago, before humans arrived to South America, ancestral populations of Solanum pimpinellifolium migrated to Mexico, acquired different traits, adaptations to a new climate along the way, and diverged into this new species, Solanum lycopersicum, variety Cerasiforme. And then tens of thousands of years later, it moved back to South America, but this time in a much shorter time span, probably carried by humans who are also carrying other early domesticated crops, such as maize. Back in South America, it hybridized with wild populations of Solanum pimpinellifolium. The native people of South America might have selected and domesticated Solanum lycopersicum Cerasiforme at that point. It then moved back to Mexico, where the native people of Mexico domesticated the large fruited tomato that we all recognize today. So we don't really have records of what early tomatoes selected in Mexico look like in terms of herbarium specimens or images. What we do have is what was characterized by the Europeans in the middle of the 1500s, shortly after the conquest of Mexico, and when the tomato and other crops were moved back to Europe and grown in botanical gardens and characterized by botanists. And what we can see is that there really was quite a bit of morphological variety. Tomatoes at this point were yellow, they were red, they may have been purple, they were different shapes, they were small, they were large. It seems like native Mexican farmers probably selected and grew and preserved a diversity of varieties with different morphologies. So shortly after that, the tomato began to spread outside of botanical gardens and into local cuisines and cultures across Europe, starting in Spain and Italy, and then moving to other countries. And as this new crop was integrated into new cultures, we saw this radiation of morphological diversity and development of novel use types. So in Italy, you see the development of types that some of you might be familiar with today, like the San Marzano paste tomato, a type with high soluble solids for processing into sauces. In Spain, there was the development of these storage tomatoes, these de Penjar varieties that traditionally were hung and stored for the winter. And what's kind of interesting or paradoxical is that despite all of this morphological variation, genetic data shows that actually these vintage or traditional European landraces of tomato have a very narrow genetic basis, much more narrow than the wild populations that preceded them. And it seems hard to believe, but if you are familiar with some of the effects of some of the mutations that are used in tomato breeding, it kind of starts to make sense. So this figure on the right here shows just the effects of 4 different genes with strong effects on tomato fruit shape variation. And you can see how different combinations of alleles at these 4 loci result in this wide variation of tomato shapes among modern market classes of tomato. So thinking about genetic diversity in tomatoes' evolutionary history, it appears that there were multiple incidences of genetic bottlenecks affecting the genetic diversity, but also a few cases where as a result of admixture and crossing with wild relatives, the genetic diversity increased. So first, as pimpinellifolium migrated to Mexico, there was a reduction in genetic diversity. But then as those populations moved back to South America, crossed with the wild population of pimpinellifolium, there was an increase in genetic diversity. After domestication in Mexico to the large fruited domesticated type that we're familiar with, there was a further reduction in genetic diversity that was made more extreme with the bottleneck as those moved out of the Americas and into Europe. But actually, if you look at the genetic diversity in modern varieties today, there's actually been an increase in genetic diversity in modern tomato varieties, which is, kind of contradicts something that a lot of us think when we think about the results of modern plant breeding. And the reason for that is because plant breeders have intentionally introgressed alleles from wild species into modern tomato varieties as sources of important agronomic traits. So here I'm showing the phylogeny of tomato species that I showed earlier. Those species that I've circled here have been used to identify traits of interest, move them into tomatoes, and those traits are used commercially today in tomato breeding. And it's not just the most closely related relative Pimpinellifolium, it's some that are quite diverged from domesticated tomato. The species that I put an asterisk next to, there's reports in the literature about QTL or genes being mapped and moved to tomato, but I'm not sure if they're used commercially or not. So sequencing data shows that these wild introgressions actually contribute significantly to the genomes of modern varieties. So if you look at these images here, the light blue represents haplotypes from modern tomatoes, and these other colors show haplotypes introgressed from wild species and the traits that they're associated with. So in some cases, if you look at chromosome 1 and you look at these processing tomato hybrids here denoted with P, you can see that they're small introgressions from Solanum habricaides that provide higher soluble solids content associated with the AGPL1 gene. But if you look at chromosome 6, you can see among a variety of modern varieties, both fresh market and processing, you'll see that over half of the chromosome seems to be represented by these two different disease resistance introgressions from Solanum chilense and Solanum peruvianum. And then that's even more extreme on chromosome 9 of tomato, where essentially all of the fresh market hybrids that were sequenced in this study have a very large introgression from Solanum peruvianum, conferring resistance to tomato mosaic virus. It's even more amazing to think about this when you think about or see in seed catalogs the extent of introgressions that are present in modern tomato varieties. So here's a screenshot I took from the Harris seed catalog. This is a tomato called Maverick, bred by an alumnus of Cornell, Jason Cavatorta, with Earthwork Seed. And if you look at what I highlighted in terms of its disease resistance package, we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, resistance to 12 different diseases or disease races. A few of these might be represented by multiple genes, and all of those are independent introgressions from wild species that tomato breeders have used in breeding. So before I go into the focus of my lab and talk about a few examples of how we're using genetic diversity in tomato breeding, I thought it might be useful to just give an overview of kind of what the tomato industry looks like in the United States and where the tomatoes that we eat come from. So in terms of acreage, the state with the most significant acreage is California, with almost 300,000 acres of tomato. And this is predominantly processing tomatoes. So this is a very highly specialized industry with specialized varieties that are machine harvested, and these are processed into ketchup and tomato sauces and all other products that we eat. The state with the second highest amount of tomato acreage is Florida. There are about 30,000 acres of tomato in Florida. Historically, Florida is where the tomatoes that we were shipped across the country and consumed in the off-season when tomatoes couldn't be grown elsewhere were produced. And historically, the way this happened was these were varieties that were picked when they were what's called mature green, so before they start turning red. Off the plant, they were exposed to ethylene gas to promote ripening. And this way, they could maintain a very high level of firmness and tolerate being shipped around the country. And that industry still continues today. Increasingly, though, if you go to the supermarket and look at the tomatoes that are there and look at the sticker and see where they came from, you might notice that a lot of them come from Canada. And these are varieties coming from high-tech greenhouses. These greenhouses, there's a large number of them in Ontario, also many in the Northeast and Midwestern United States. And this is another very highly specialized production system where these greenhouses grow indeterminate or vinding types. They're plants that are grown for 10 or even more months. By the end of the season, these things are dozens of feet long, and these are producing pretty, high quality tomato varieties that are sold often, at retail in supermarkets in clamshells. Finally, the last major kind of production zone I'll mention is Mexico. We import a lot of tomatoes from Mexico. Unlike Florida, these are typically harvested vine ripe. Many of these varieties have a gene that offers extended shelf life. So they have slow ripening and they can tolerate being shipped when they're ripe. And you'll see a lot of these in the supermarket as well. So I would say those are the four major production regions. And what I didn't mention was New York in the northeastern United States. And that's obviously where we are, and that's where my work is based. So in New York, we have around 2,000 or maybe a little bit more acres of tomatoes grown in the open field. There's probably similar acreage in Pennsylvania and New England. So compared to, say, Florida or California or Mexico or high-tech greenhouses, the total value of that seed market is more minor, right? So in terms of private sector investment in breeding, there's fewer resources devoted to breeding for this region. But it's still a significant amount of acreage, and because farms on average in the Northeast are smaller than many other parts of the country, collectively, this is representing thousands of individual vegetable farms. For many of these people, tomato might be a very significant crop in terms of their profit margins, and there's still many needs that need to be addressed for this area. So, what are those needs? Things that I would say would define tomato production where we are is, we have a pretty short growing season, May to September. We have warm, humid summers that result in high fungal and bacterial disease pressure. And those diseases that we have in the Northeast are, in many cases, different than the diseases that are of importance in the Southeast and in protected culture environments. In the Northeast, we also have very high proximity to major population centers, people with very high demand for good quality produce. And that results in farms having a diversity of marketing strategies, people selling wholesale and people selling directly to consumers through things like farmers markets and community-supported agriculture. So in terms of my lab's research and extension goals, to summarize it at a really high level, what we do is we try to release cultivars and germplasm adapted to the northeastern United States with high yield, quality, and disease resistance. We discover the genetic basis of traits of agronomic, or in many cases, traits that are important to the Northeast, but also to other production regions. Another goal we have is to model strategies for the implementation of new technologies and applied vegetable breeding. And then as part of our extension mission, we try to generate and disseminate results on variety performance in New York for our stakeholders, which are farmers, gardeners, and members of the seed industry. So I'm now going to get into two different projects that we're working on, leveraging genetic diversity from tomatoes past at kind of different places in its evolutionary history. So for the first project, we're accessing diversity that diverged from tomato millions of years ago, and this is from the species Solanum sidians, where we're looking at sources of abiotic stress tolerance. And then, instead of millions of years ago, we're also accessing diversity that diverged from modern tomato maybe more like 100 years ago or 150 years ago. So this figure is definitely not to scale. And that's quality that we're trying to access from heirloom varieties for modern tomato breeding. So I'll talk a little bit first about Solanum sidians. Solanum sidians is a tomato wild relative that grows in the Atacama Desert of northern Chile. The Atacama Desert is the most arid non-polar desert on Earth. On average, it receives about half an inch of rainfall a year. And some weather stations in the Atacama Desert have never recorded any precipitation as long as they've been there. But what's remarkable is if you look at these pretty desolate landscapes, there are these wild tomatoes that are able to grow there. So it seems like a pretty remarkable source of tolerance to these very harsh conditions. If you look at the phylogeny that I showed earlier, you'll notice that I didn't have a circle around Solanum sidians. And that's because for a long time, breeders weren't able to access genetic diversity in Solanum sidians because of reproductive barriers with Lycopersicum. You can't cross Lycopersicum and Solanum sidians. However, Roger Chatelet at the University of California, Davis, over the course of many years, decades actually, completed a very complex series of crosses using bridge species in order to move genetic material from Psidians into Lycopersicum and developed a population of introgression lines. These are 56 lines. Each of them have a single introgression from Solanum Psidians in the background of domesticated fresh market tomato. These introgressions range in size, but on average, they're about 20 megabases, which, most of them probably have several hundred genes. And this population was developed and released recently for use by the tomato breeding community. So we were very interested in this when I first started, and we still are. There are multiple traits that we're interested in looking at. One of them is salinity stress, and there's a few different reasons why. So salinity stress can depress tomato yield in a few different environments of commercial relevance. So in the Central Valley of California, where, like I said earlier, there's almost 300,000 acres of tomato, there are actually millions of acres of cropland there that are affected by pretty high groundwater salinity. This red area shows areas where the salinity is so high that there's a yield impact on tomato. In New York, we're a much wetter climate. Soil salinization is not really so much of an issue. But actually in certain kind of production systems, it can be a problem for farmers. So in recent years, in the past maybe 10, 20 years, the production of tomatoes in high tunnels has really kind of exploded in New York and in many other states. And what happens when you put some sort of covering over your plants and preclude precipitation from falling on the soil is you prevent leaching of salts that build up through application of fertilizer and your irrigation water. So this production system actually results in creating something more similar to like an arid climate as in California. This can be managed through cultural practices, but still in recent years we've seen many farmers struggling with high levels of soluble salts in their high tunnels. So there have been populations of Solanum Psidians found growing in basically like salt basins, soils with an electric conductivity of almost 100 decisiemens per meter, which is around 40 times the level that tomato can tolerate. The particular accession that was used as the parent of the introgression line population, however, hasn't been characterized for its salinity tolerance. So the first thing that we wanted to do was verify that it's tolerant to salt, and also develop some methods or protocols for evaluating salinity tolerance. So this work has been led by our lab's field and greenhouse manager, Howard Rice, who has been very creative in designing the irrigation systems that I'll be showing and leading these experiments. In the literature, there's many different ways that salinity is evaluated in tomato. Oftentimes people evaluate it in terms of its impact on vegetative biomass at the seedling stage. We really wanted to look at fruit yield, the most directly relevant agronomic trait. So the way that we did that was we grew plants with drip irrigation, and then with these fertilizer injectors, instead of injecting just fertilizer into those irrigation systems, we injected table salt as well at different concentrations. We grew the plants to maturity and evaluated their yield. I'm just gonna pull my phone out to get a quick time check. Okay, I think I'm good. So in this first experiment, we looked at 4 different genotypes of tomato. The 2 parents of this introgression population. So this fresh market line, LA4354, the Psidians donor here. We also, as a susceptible check, we looked at a processing tomato hybrid. And as a resistant check, we looked at an accession of Solanum pinellii that's been characterized by others for salinity tolerance. Most of that work, though, has been at the seedling level. In this experiment, we grew the plants under 4 different treatments, a fertilizer-only treatment, and then 3 different levels of sodium chloride, up to a total electrical conductivity of 17.5 decisiemens per meter, which is much higher than what you would, any farmer would hope to find in their soil. So here I'm showing the effects on fruit yield for the 2 tomato entries in this experiment. If you look at the red, that's denoting the fresh market parent of the introgression line population. The green is the processing tomato hybrid. you can see on the left here a pretty steep decline in yield with increasing salt concentration. Under the highest level of salt that we applied, the yield was maybe around 20% of what we saw under the control. You can also see that the most precipitous decline was from the control to the first level of salinity applied, and after that it kind of plateaued. Something that's interesting that we haven't really followed up on, but seemed to be a curious result was, although the response with these two different varieties was the same in their total yield, the way that that was realized was different actually. So Solanum, the fresh market tomato variety, showed a decrease in fruit weight, whereas the processing tomato hybrid showed a decrease in fruit number. But that resulted in the same response in terms of total yield. So for the two wild species we had in this experiment, we can't evaluate their fruit yield because they devote a really negligible proportion of their biomass to fruit. So the way that we looked at this was we looked at their vegetative biomass. And the results of this were kind of surprising to us. So if you look at, again, at the domesticated tomato genotypes in this experiment represented in orange and green, you can see that there wasn't really a strong response to salinity in terms of their vegetative biomass. In fact, under 2 of the salt dosages, the fresh market tomato line showed greater vegetative biomass than under the control. The 2 presumably resistant wild species actually showed stronger declines in vegetative biomass. If you look at Sidians, which is this teal color here, you can see the difference from the control to the highest salt concentration there. In terms of the absolute difference, it was fairly minor, but that's because Solanum Sidians we found to be a slow-growing plant. There wasn't really much biomass to decrease. But as a relative difference, actually it had the steepest decline of all 4 of the entries in this experiment. So that was kind of like a disappointing result, right? You don't want the donor of the trait that you're hoping to introgress to show a poorer response than the parent you're hoping to introgress it into. But after thinking about it a little bit more, maybe we decided maybe it's comparing apples to oranges, right? We're comparing these species who don't produce any fruit, they don't devote any energy to fruit production, and comparing their vegetative biomass to tomato varieties that do devote a lot of energy to fruit production, and we saw that fruit production was affected. So we moved forward with screening the introgression lines, thinking that maybe in the background of domesticated tomato, we'd be able to see more of the effects of the Solanum Sidians genome. So we started by looking at 27 introgression lines. Not all of the complete set was available for dissemination when we started this experiment. We also avoided lines carrying introgressions in a heterozygous condition. These are introgressions where there's a recessive lethal mutation in the background of domesticated tomato. And to make our lives easier, we just avoided working with those. We evaluated these under both control conditions and a fertilizer treatment with about 75 millimolar sodium chloride. We completed the first replicate of this last fall, and the second replicate is we're hoping to wrap up in the next month or so. And here, as opposed to looking at the wild species itself, here we were able to see an effect of salinity tolerance when you look at the gene effects in the background of domesticated tomato. So in this plot here, on the left, I'm showing the distribution of fruit yield under the control. On the right is the distribution of fruit yield under the salt treatment. There's lines connecting the performance of individual genotypes between those 2 treatments. The red line is the domesticated parent, recurrent parent of the population, and the black lines are all introgression lines. We saw, unsurprisingly, that the domesticated parent did pretty well for yield under control conditions. It ranked 5th, but under saline conditions, it ranked 17th in terms of its yield. And then in terms of the severity of its percent decline in yield, it ranked 3rd. So out of all the entries, it showed the 3rd steepest response in terms of its difference in saline versus control conditions. The introgression lines ranged from a response of about a decline similar to the domesticated parent, around 80%. And the best introgression lines showed a decline in yield of around 45%. We also saw that there were 4 introgression lines that out-yielded the domesticated parent under both control and salt conditions. So what are our immediate takeaways from this experiment? Well, for one thing, by the fact that almost all of the introgression lines did better than the recurrent parent, it seems like there are multiple genes distributed across the genome that provide partial effects individually on salt tolerance. My background is in disease resistance breeding and pathology, right, where we can identify single genes, R genes, with major effects. And I probably should have done my homework and read some more about plant physiology. This might not have been such a surprising find in terms of abiotic stress resistance. So this leads to the question, are these genes, do they have effects that are large enough to be commercially relevant, right? We can look at the lines that did the best, right? They had a 45% decline in yield compared to an 80% for the domesticated tomato. And you can look at that 2 ways, right? One is 45% is much better than 80%, but also a 45% decline in yield is still a 45% decline in yield, right? Is that enough to be relevant commercially? So we need to do further experiments to figure that out. One thing that we're interested in doing is combining different introgressions that each had pretty, good effects individually and seeing what they look like in combination. We're currently doing a crossing block to try to answer that. And then another question that we need to answer is, can we reduce the sizes of these introgressions enough to get rid of linkage drag while retaining the effects on salt tolerance? So there's a lot of work to do, but we're excited about this project. Just looking at the fruit yields from the control on the left to salt on the right, except the pattern's actually reversed for this lower picture. I don't want to mislead anybody. You can see the variation in response among these lines, and visually it's pretty observable, the impact that these introgressions have. We're also moving on to look at other traits that we're interested in. One of these is freezing tolerance. The accession of Synapsis that was used as the parent of this population was characterized and discovered growing at really high elevations where the temperature goes below 0 degrees. Pablo Betancourt is an intern in my lab who's leading this work. And we've been really excited to see that when you throw tomatoes in a growth chamber at pretty cold temperatures, they come out looking like this. But when you throw Synapsis in there, it comes out looking like that. It seems like maybe a more far-fetched project than even the salinity tolerance project. But what we hope is that we can develop tomato varieties that have freezing tolerance as seedlings. Farmers can transplant them to their fields maybe a few weeks earlier than they typically would, get season extension, maybe harvest tomatoes earlier in the season when prices are better, and not have to worry about late spring frosts. So Pablo just this week, I think, wrapped up screening the rest of the introgression line population. So if you're interested in the results of this project, come talk to him during the event today. Okay, so I want to move on and talk about another project that we're doing focused on using heirloom tomato varieties in breeding. So I want to give a little bit of a historical perspective. So in terms of modern tomato breeding, so breeding trends in the 20th century, what those trends reflect are kind of larger ongoing trends in society at the time, right? Industrialization of agriculture, the commodification of produce, and that's reflected in the traits that breeders were selecting on. Things like uniformity, firmness, fruit firmness and shippability, fruit size, shape and color, disease resistance, yield. I don't have on there flavor. It probably was not a major trait under selection for a lot of this modern tomato breeding. Towards the end of the 20th century, so starting maybe in the late 1980s or 1990s, the heirloom tomato started kind of growing in popularity. If you look at this figure on the left, This is showing the mentions of the term heirloom tomato in two newspapers from 1989 to 2005. And you can see that, at one point it was something that didn't really exist in our popular consciousness. And within a few decades, it became something that was common to talk about. And that was the result of maybe a few different larger movements or trends happening in society that kind of represented maybe a pushback against some of the industrialization of agriculture that was happening at the time. So the Seed Savers Exchange was established in 1975. That's a network of individuals interested in preserving seed that maybe was handed down to them in their family or in their community, saving those varieties and distributing them among a network of other seed savers. Chefs like Alice Waters opened restaurants that were the first to champion what we now refer to as farm-to-table cooking. They included items on their menus like heirloom tomatoes. And, pretty soon heirloom tomatoes became ubiquitous at farmers markets, upscale restaurants, and today they're pretty common to find in grocery stores. At Wegmans here in Ithaca, I can see them all year round grown in greenhouses in Canada. So what exactly is an heirloom tomato? And the answer is, we don't really know. There's no scientific definition for this term heirloom tomato, but there are a few different definitions that are thrown around, more popularly. So here are some of the most common ones. They must be open pollinated. They must have been introduced more than 50 years ago. That's kind of an arbitrary cutoff, but what it means is that these aren't the results of modern breeding, right? These are heritage varieties that were selected in a previous time. And then according to this book, Taylor's Guide to Heirloom Vegetables, they must have a history of their own. And many of these varieties are accompanied by stories, like familiar lore about where these came from, who originally selected them, how many generations they were passed down, that make them kind of more interesting. I think if you do ask most people what an heirloom tomato is, maybe I'll poll all of you later, I think they probably wouldn't refer to these things. Instead, they'd probably be thinking of visual traits, right? Things that differentiate them from round red tomatoes, differences in size, shape, and color, maybe some defects that have been selected against defects, depending on who's deciding what a defect is. Things like green shoulders or scarring on the blossom end of the fruit that are all associated with these varieties. So these are a few of my personal favorite heirloom varieties. These are also ones that are among the most popular and most well-known. Cherokee Purple, Brandywine, both pink Brandywine and yellow Brandywine, striped German. If you've grown these in your garden, maybe you'll agree with me that these are very delicious varieties of tomatoes. They're beautiful, and their quality in terms of eating them fresh in a salad is really excellent. So I think, part of the popularity of heirloom tomatoes is not only for their cultural associations and what they mean in terms of self-sufficiency and seed saving and a pushback against industrialization of agriculture, but also for their value in and of themselves as really excellent varieties. So in recent years, there's been kind of a movement with some seed companies to develop varieties combining some of these qualities found in heirloom tomatoes with traits more typical of modern hybrids. So things like disease resistance, resistance to cracking, high yield potential. This is a screenshot from High Mowing seed catalog. And one of the terms that some of these companies is using is highlooms, a combination of hybrid and heirlooms. We like that term a lot, and that's a term that we're using in our lab to describe the breeding efforts that we're using. One thing that we started doing in the lab was variety trials of commercial high-loom varieties. Here you can see the harvest from these varieties in bins in preparation for a field day we had last summer. You can see the variation in color, how beautiful some of these varieties are. In terms of the data that we collected, we also saw that there have been pretty significant improvements in terms of many agronomic traits. So this plot on the right is showing these varieties in terms of their percent grade 1 fruit. So the proportion of fruit that can be marketed is USDA grade 1, meaning free of blemishes and other defects. If you look at Brandywine Pink, which is our heirloom check, the percent grade 1 was about 25%, which, if you think about that from a commercial agriculture perspective, that's pretty bad if a farmer has to discount or throw away 75% of the harvest. But these highland varieties, get up to 70%, so a pretty big improvement. We also trial just normal standard slicer tomatoes. We found in our trials that the percent grade 1 on those can get up to, say, like 95%. So there's still probably are trade-offs in terms of things like resistance to fruit defects, shippability, and traits relating to eating quality, softness, texture, thickness of fruit skin, etc. So another thing that we did when I first started in 2023 was surveyed growers in New York about their perceptions of both heirloom and highland tomatoes. This was in collaboration with Teresa Rusnick, vegetable specialist with Cornell Cooperative Extension. And out of the 76 tomato growers in New York who responded to our questionnaire, we found that about 3/4 of them grew heirloom varieties as a portion of their production. So that kind of speaks to the demand that's grown for this type of product. Fewer grew highland varieties, about 44% grew highland varieties. And when we asked what the challenges that they found associated with heirlooms, there were things that we weren't surprised by, disease susceptibility to fruit defects, and poor storability or shippability. What I found more interesting was when we asked, what are your desired attributes in terms of future highland varieties? And when they ranked these, the top 4 were: number 1, resistance to diseases common of the open field; number 2, resistance to fruit defects; 3, determinate growth habit; and 4, resistance to diseases more common in the high tunnel. And the reason that this is interesting to me is that most of the seed companies currently are breeding types that are indeterminate and maybe more adapted to the high tunnel or the greenhouse in terms of their disease resistance package. So it seemed like there were some needs that still may be left needing to be addressed. So we started crossing modern varieties with heirloom varieties in 2023. In terms of the modern lines that we're using as parents, we're making use of lines developed at Cornell by my predecessor, Martha Muchler, who developed lines with a really comprehensive disease resistance package to diseases in the open field in the Northeast, including several novel disease resistance genes in QTL that her lab discovered. And then we crossed a few of these lines with a number of heirloom varieties. I have 4 listed here. These are the populations that we carry forward in the scheme that I'll show you in a few slides, but we made crosses with many more. And the objective of this work is to develop attractive, crack-resistant, high flavor lines with strong open field disease resistance and determinate growth. We've been following a pretty aggressive timeline to move this project forward. So in the winter 2023 and 2024, we carried out 2 generations. We did the crossing block. We did the F1 generation in the greenhouse. The F2 in the field last year, we used marker-assisted selection to enrich those seedlings for the disease resistance alleles, especially for the diseases that we were most interested in. We then made visual selections in the field for fruit quality traits and for determinate growth habit. So that's a determinate growth habit is a concentrated flowering type that sets flowers in a short window and then kind of stops growing. The selections from that field here, a picture of a couple that we're really excited about. If we continue to see fruit like this, we'll be very pleased. And we sent those to Winter Nursery in Costa Rica for single seed descent to the F4 generation. And this year, we plan to get back around 320 F4 families from this work. These are derived from 10 crosses, these 320 families. Not all of them are part of this high loom breeding project, right? We also have crosses that are, I guess you would say, modern standard varieties by modern standard varieties, but 6 of them are from heirloom by modern crosses. And one thing that we're excited to do with this population is use it as kind of a test case for how we might be able to implement genomic selection in our tomato program. So this work is being led by Jack Fabrizio, who's a postdoc in the lab. And the specific strategy that we're planning to use is what people call a test and shelf strategy. For us, one of the biggest opportunities that genomic selection poses to a vegetable breeder is the opportunity to have our phenotypic data stretch farther. Collecting data like yield or fruit quality on a perishable crop that has to be picked multiple times is really labor intensive. It's very difficult for us to do that in a large number of entries. But if we can do it on a certain number of entries, then predict into a larger number, we think that can have a pretty big impact on our breeding program. So this year, out of these 320, we're going to put 80% of them in the field, genotype those, phenotype them as well, and then we're going to predict on the entire population, including the 20% that we're only genotyping and not phenotyping. So here's a kind of a quick schematic of what that would look like. So those 80% will be used to calculate genomic estimated breeding values, including on the holdout set. So among that entire population of 320, we'll select the top 10% across the entire population. We'll carry these lines forward for further product development, further testing and breeding, maybe creating some test hybrids. And then we'll also intermate those, complete another cycle of modified single seed descent. And then we think that as we continue to do this and generate more historical data, we can change the proportions, right? So maybe in 2 years, when we have another population of F4 lines, instead of phenotyping 80%, maybe we can phenotype just 20%. And we can include a larger population, and we can use some of the historical data as well to make predictions. There are lots of questions to answer on the practical implementation of genomic selection, not just in my program, but I think personally in vegetables in general. There's literature answering these questions in many agronomic crops that haven't been addressed really in many vegetables. So for us, we're really interested in how well can we predict within families, across market classes. Are these high loom crosses going to be predictive of standard tomato by standard tomato crosses? How many markers do we need? Is 1,000 enough? 1,000 is kind of the number that is financially reasonable for us. So we're hoping that's enough. What training population sizes are needed to achieve reasonable prediction accuracies, right? Like I said, it's a lot of work to collect this training data. How much do we really need to collect in order for this strategy to work? And then tomato is a crop that's primarily marketed as F1 hybrids, even if the inbreeding depression is not as strong as in a crop like corn. We're going to be predicting the per se value of these lines. Is that the right strategy? Should we instead be predicting general combining abilities or specific combining abilities? These are things that we're hoping to figure out. So I'm going to start wrapping up my presentation. I think one thing that's kind of in common across these two projects that I described is the fact that these traits that we're interested in are quantitative traits. So the history of tomato breeding, there's been tremendous progress in trait improvement, focusing on moving qualitative major effect genes from wild relatives. In terms of using similar approaches to improve quantitative traits, it's probably going to be a bigger challenge. The way that we're doing that with these high loom breeding populations, we think genomic selection might help, right? We're using breeding methods that we think are suitable for the improvement of quantitative traits. We don't expect necessarily to find amazing varieties in that first cycle. We might need a few generations of recombination to break up linkages between alleles. But we think that using this approach, we can develop high quality lines. In terms of the work we're doing with Solanum sedians, where we found that there are multiple genes conferring resistance to the stress tolerance that we're interested in, the linkage drag is probably so extreme that there's only a certain limitation in terms of how many of the introgressions we can practically use in commercial breeding, right? Maybe there's a world in which we can use 2 of them or even 3 of them if we're able to reduce the size of those introgressions sufficiently. But beyond that, it's probably not feasible from a practical point of view. So I think we probably need creative solutions moving forward. I don't know exactly what those are. Maybe I'll be inspired by the rest of the talks today. Maybe there's a world in which we can use these genetic resources, these populations that have been developed, the phenotyping platforms that we're using to identify the genetic basis of these traits. And then perhaps instead of actually introgressing them into tomato, we can just make the edits in tomato through genome editing, corresponding to those genetic differences that lead to the phenotypic change. So with that, I'll wrap up. I talked about the work that some of my lab members are doing. I didn't have the chance to talk about the work that my students, Ivana Castillo and Diana Mulder, are doing. They're doing really exciting projects in disease resistance genetics in tomato and eggplant. I also want to thank the work from former lab members who have contributed, especially to a lot of the field work that we've done, and then the funding sources that we've had for the projects that I've described, including the California Tomato Research Institute, who's funding some of the work with Solanum pseudians and salt tolerance, USDA federal capacity funds, Cornell, and the Vegetable Breeding Institute. So if we have time for any questions, I'd love to answer them, and I'll also be around the rest of the day, and I look forward to talking with some of you then. Thank you. Yes.
More about your process for honing in on your breeding goals and focus for this program?
Sure, that's a very good question. So the question was, what the process was for honing in on our breeding goals. I think it was through a combination of things, and I mean, we're still working on them. For me, the most important thing is to be out there and talk to people, learn what problems are, learn what needs to be addressed, learn what solutions already are out there and don't need to be reinvented from scratch. So, my first year, especially my first summer in my position, I tried every week to visit at least one different farm, right? I traveled to different parts of the state, got in touch with extension specialists in those regions, got introduced to vegetable growers in those areas, just learned about what they did, asked what varieties they liked, why they liked them. If you could have your ideal variety, what would it look like? And those conversations really helped contribute to a lot of the work that we're doing and how we're moving forward. Yes. Hey, Lara, thank you.
Really cool to see all the work that you've been doing, and I love this idea of islands. And my question is, so you're looking at a lot of the agronomic traits, but I'm assuming a lot of the motivation of using the heirlooms is to introduce some of those more, unique flavor and taste profiles, but that's of course a really hard thing to quantify. Do you have any plans to incorporate that into your genomic selection?
That's a really excellent question. The question was, how do we plan to select for flavor in this high-loom program? And you're right, that it's really tough to select for flavor. In our early generation nurseries, we walk through and we do a lot of tasting, but we get very sick of tomatoes very quickly. And I'm someone who loves tomatoes. I never thought that would happen to me. So to start, we're looking at some things that are relatively easy to quantify: Brix, acidity. We're going to do some taste testing too, not formally, but informally, and see how well those traits are associated with our informal assessments of flavor. And we're going to go from there. I mean, tomato flavor there's been a lot of literature on what controls our perceptions of tomato flavor, and it's a mixture of sweetness, acidity, and then lots of different volatile compounds. And measuring those is obviously a lot more work. So we need to see how we can balance our resources and do that efficiently moving forward. Yes, Lance.
I got two questions. It always strikes me that we've taken this perennial shrub and we've kind of imposed this annual lifestyle onto it, especially with the wild relatives. Are we evaluating the traits of these things too early in that first season? And if we were able to give them time to establish, do you think they would be more resistant to some of the stressors? And the second question is, given those big introgressions and how divergent like Scythians is, Roger probably couldn't break those introgressions up with crossover and stuff. So how do you plan to go about breaking these things down?
Okay, yeah, thanks you. Those are good questions. Yeah, so I think the first question was, how do we evaluate phenotypes on Synapsis when it's probably a perennial and its growth habit is so different, And I think the answer is we probably didn't really do a good job of doing that. We grew it in, almost hydroponically in a greenhouse, the way we grow normal tomatoes. This is a thing that's adapted to the desert, right? We put in a very different kind of artificial condition, and probably not the best way to assess the traits that we were interested in assessing. However, when you look at the effects of portions of the genome of Synapsis in the background of domesticated tomato, I think it's much easier to kind of isolate and evaluate those traits. And your second question was, how do we plan to break apart those recombinations? And we don't know how easy or difficult it's going to be, right? The way that we are planning to do it now is to just try it and see what happens. Maybe that's magical thinking, but I guess the only way to find out is by doing it. Yes.
Thank you, Greg, for that great talk. I was just wondering, you mentioned that heirloom varieties have been maintained by seed savers, and that kind of came out of a response to modern agriculture, modern breeding. So how do they feel about hylims? Is there acceptance within the existing heirloom community? Are they excited about disease resistance, or are they kind of not liking this?
That's a really good question. The question was, people who are really attracted to heirloom varieties for various reasons, how do they feel about hybrid versions of those heirlooms? And I think there's a diversity of opinions. I think people who are interested in heirlooms primarily because of their flavor and appearance are open to new varieties, even if they're hybrids. There are other people who have political opinions on F1 hybrids and the control of seed, intellectual property protection. Those people probably aren't as thrilled about the development of hybrid varieties. But these heirloom varieties, the point at which you can only get them by writing a letter to somebody else who saved them, that was a long time ago. These varieties, seed of them are sold by all sorts of major seed companies and grown commercially now.
That's hard. So that's all the time you have for questions. So please give Greg a round of applause.
Awesome. Okay, so I have the honor of being, I think, the side of this symposium that's more about conservation, which you might see, or you might remember is in the title itself, Conserving and Utilizing. So it feels like an honor and also a responsibility to bring the other half of the equation to what seems like a really strong and wonderful community, mainly of plant breeders, genetics folks and such. And so a great community for me to connect with and to speak with. Thank you for the invite to the graduate student organizers and to Cornell and to Corteva. Really appreciate it. And I do not take for granted opportunities to get together and collaborate, especially in this day and age. And so thank you. My goal, I think, above all else with this talk is to engage you, enroll you even in how important that side, the conservation side, is to your work. And so let's see how I do about that. This is a real photo, by the way, not just a screensaver. This is from the Great Sand Dunes National Park in Colorado, and that's a wild relative that I'll talk a little bit about a little bit later. Yeah, so I'm going to be talking about things like this flower on the left side and its relation to the flower on the right side, and how those two are still related after so many millions of years. Just as a reflection, when I started in this work like 25 years ago, I do feel like we were all the weirdos that they didn't know what to do with at the Tri-Societies meetings. We had sort of our own little group that nobody came to, there's 4 people in the audience and all of that thing. That was the crop wild relative people. And it's not that the plant breeders weren't using the wild relatives. They certainly were and all of that, but it was much lower on the awareness screen there, I think, at the societies and also in the conservation world. And that's not true any longer. There has been this huge sort of shift, as I've seen it, into public awareness, like all of these articles around wild foods or wild relatives or genetic resources, even terms like that, that you never thought might end up in the news. And You might have seen that the World Food Prize this year was given to conservationists, including of wild relatives of crops, and all kinds of crazy things are happening in the world with wild relatives. And that's true not just of public awareness, but even in our very largest global or international accords and agreements. For instance, the Sustainable Development Goals and the new agreements and the older agreements of the Convention on Biological Diversity. These are really our largest international instruments of countries agreeing with each other about biodiversity, about food and agriculture. And they're all saying things like wild species and wild relatives in them, which is wild in and of itself. That actually was not an intentional. So I have a couple of slides that are a little bit simple for you all in this group, but I will put a few definitions out and a little bit of the history. For me, I keep it really simple. Wild relatives are wild plants that are related to our domesticated crops. As you saw from the tomato group and from the brassica talk and others, the exact definition or which plants are in and which plants are out for each crop varies a lot. And the wheat talk coming up after me is another example of a different way of defining the edges of the wild relatives. The theory behind this goes back 50 years, 53 years now, to some science that was really based mostly on crossing, on breeding studies and seeing what could cross with one another, and coming up with this kind of onion concept of the center being the crop and its related species, same species that's wild, and then as you go out from the onion, it gets less related and more difficult to cross. These days, those boundaries have really fallen through modern tech, and so you can double chromosomes easier, and you can do these bridges with other species, as mentioned in the brassica, and all these things. And so the edges of the gene pool 2 and 3 and out from there are getting fuzzier over time. If you're interested in the picture of like who is a wild relative for what crops you're interested in, there is a wonderful database, probably the only like truly global database run by USDA GRIN Global Taxonomy with that link there. I hope these presentations are available to everyone after this. I'm happy to make mine, but there's a lot of links in this presentation if useful to you. You can go there and you can put in your crop of interest, and it will start to tell you based on literature who are the wild species and what have they been used for that has been published. There's been a couple of reviews of the uses of wild relatives over the last 50 years or 100 years in this case, and essentially the trends are there's more and more publications, folks saying that they're using wild relatives. And again, this is not the private sector so much, this is what's being published, and so it's even higher the number of uses, of course, when you get into the privates. And then as you might have seen already from the talks we've had, the major uses so far are primarily pest and disease resistance, also abiotic stress tolerance, and then many other things. Two final points as we just get into it about wild relatives that I like to make. Again, you all as a very educated crowd will not be surprised by this, but the complexities of their use are wondrous, and it's there is of course still just bringing in single genes for important traits like pest resistance, but then there's much more, I'm not saying any better or worse, but much more complex versions like grape. And so what grape is, for those who don't know, and I know this is a big crop in New York, grape has a top, the scion, that can be different than the bottom, the rootstock. That top scion is still mostly in most places in the world one species, the domesticated species vinifera, but that is not the only species that's out there. And in fact, there's more and more coming along of hybrids with vinifera and other things. And there is a case, this is a rather extreme case of this Chambourcine, which is, we don't actually even know all the different species in it, but you can see the list of 6 or actually 7 different species actually in this hybrid that's grown on the East Coast here. And then the rootstock there is now almost by standard or by definition, one of 3 or 4 different wild species from North America. That's how grapes are really able to grow around the world for the most part. And so This is the landscape really of wild relatives and of how much they're used. They're really, really integrated into some of our most major crops. And I had to put this picture up here for several reasons. One is because this is Cornell work, seminal work from 25 years ago. That was one of these papers that really did start to raise the awareness and change maybe people's minds, even breeders' minds about using wild relatives. And so Dr. McCooch, who's maybe listening in online, this is a kind of a synthesis paper, and it was making several points. And one is that you don't always know what's in these plants, which is the wonder and also the challenge of them. And so the photo here, the A was the wild relative, which was spoken about by our first speaker here. What a wonderful presentation. Thank you. I learned so much. The B on the left side there, that was the domesticated version before the introgression. The right side of that is actually after the cross with this green tomato. You get a darker red version of the tomato. So that darker trait came actually from the wild green relative. And then on the bottom side on the C, this is a smaller one. This is, I believe, Pimpinellifolium crossing with that medium-sized tomato to get even a larger tomato through, kind of through that vigor effect. And so you don't always know what you're going to get. Okay, so the talk that I am going to give today also, as much as it's about plants and about genes and conservation, it's really also about people. Conservation really can't happen by an individual or an institution. It's a collaborative effort. The bigger and more collaborative it is, the more it works. Relationships matter to conservation tremendously. And so there was a big first step 15 years ago through a mega global project called the Crop Wild Relatives Project that was 50 million euros through the government of Norway. And that enabled giant steps forward in trying to understand who are the wild relatives of our major crops, how well are they conserved in seed banks or gene banks around the world, and I'll talk a little bit more about that in a minute, and then to start to do more pre-breeding around those. This was an important project for me because I got to do my PhD as part of it, and so I'm very thankful for that. And I think it's still having its major effects around the world. Many institutions, including Cornell, were part of this project. A little bit as part of that project, on the far right side, one of my chapters of my PhD was starting to just try to understand who are these resources in different countries. And so we published an inventory for the United States of what is this diversity. And I'm going to be talking mainly about North America in this presentation. So I wanted to list that Mexico came along with their version, also in crop science a couple of years later, and then the Canadians a couple of years later. And so we have wonderful now resources to just understand lists basically of all these species. And this is not it. There's probably 15 or 20 other countries that have now published national inventories for their wild relatives. And so that's a really neat baseline of understanding what's out there. And so to give you a short idea of who's out there, we have this pie chart that gives you a little sense of how big the slice is that are related to these major crops. And this is the narrowed down, like most prioritized set of species that we prioritize, basically based on how important the crop is. And so we've got a lot of blackberry and blueberry relatives, and grapes and pecans and peaches and plums and sunflowers and other things there. And so I would like to talk actually for a minute about why we have diversity of crops in North America, which I'm just going to talk about the United States and Canada, when not that many crops were domesticated in this area. So this is a map I worked on a while back that it's certainly not every crop in the world, but it's most of the crops that we eat. It's about 80 or 100 crops. And let's just call it where they came from, if you allow me that. I know you're a very educated crowd, but where more or less they arose, the region part of the world, and where their genetic diversity is richest and where their wild relatives tend to be richest. And you can see a few things here. One is that plants were domesticated in many parts of the world. So were animals, not just one place. I believe I was taught in high school, I'm still angry about this, that agriculture arose in the Fertile Crescent, and that was the whole story. It's not the whole story. So actually many dozens or even hundreds of places, crops were domesticated by hundreds or thousands of cultures. And that's typically understood to be maybe 28 to 32 kind of very rich centers where a lot of innovation was going on, typically 4,000 to 12,000 years ago. You can also see that North America is not the biggest list. It's kind of a smaller list. This is, again, not the whole list of crops, but there's blueberries there, and those currants or ribes, grapes, some pumpkins, sunflowers, strawberries, pecans, some other things. So I have 4 reasons I'm going to try to argue with you of why we actually have this giant inventory of 5,000 species that are related to crops here in North America. And the first one is because we do have these native domesticates. That is, people that were here in this part of the world were checking out the wild plants and realized that some of them were really awesome and started growing them and domesticating them, and they got bigger and more useful and more edible, and their wild relatives are still here. So that's number 1. We got that? Number 2 is that there is a very rich diversity of crops that were domesticated south of here, Mesoamerica and further south. That's corn and other types of squash and chili peppers and potatoes and tomatoes and many other things. And they have wild relatives, cousins, northern cousins, ones that live up here in more temperate regions but are related to those crops to the south. And so, for instance, Triticum, which is a wild relative of maize, and there's a researcher of that here, Dr. Denise Kostic, in the crowd. And so that's our kind of, let's call it there's versions that are more temperate of maize that are up here. That's number 2. Number 3 is that as a temperate region, we also have cousins or wild relatives that are cousins to crops that were domesticated in other temperate regions of the world, in Europe and in East Asia. And so there's a lot of wild plums and apricots and almonds and things like that were, the domesticates were domesticated elsewhere, but the relatives are also here. And number 4 is that we have weeds, invasives that come from other places. And those are problematic as weeds, things like in the West where I'm from, wild oats or wild beets, wild lettuce, but they are also genetic resources and they are also crop wild relatives, and they can be used for breeding as well. So those are the 4 reasons, my major 4 reasons why this is actually a very rich part of the world, even though other parts of the world are just as rich in this incredible flora that is useful for people through plant breeding. And as we'll talk about at the end of this presentation, for other reasons too. So we have been using wild relatives for a long time that are native to this region. This is just 4 examples of real uses of our native resources and wild relatives in sunflowers and in filberts or hazelnuts, in maize, and in walnut. The other side of the story, and in my contractual obligation as a conservationist to start really into this conservation story here, is that unfortunately a lot of those species are threatened or endangered, and these are all 4 of these are listed under our Endangered Species Act. Many of the species are not listed, but these are lucky enough, I suppose you could call it, to be listed and regulated there. And so, what do we do about these species that are useful to us, that are wild, that are threatened? One of the things that we do is we put them in seed banks, or gene banks, as we like to call them. And this is a picture from the National Laboratory in Fort Collins, located in Colorado State University. It's a USDA facility. The full term is National Laboratory for Genetic Resources Preservation. This is the national backup repository for the United States system. And our previous speaker who spoke about the use of the brassica collection from USDA, this would be where the long-term collection that's kept for decades or hundreds of years is kept to make sure that it stays alive and is around and can be replenished out into the more active sites. The active sites, I'd like to talk about this system just for a minute, if you will. The active sites in the USDA system are all across the United States, and that makes sense because different crops grow in different parts of the United States, and the desire is to have these active sites really working in the right ecologies. That system is the largest, probably national system in the world in terms of how active it is in doing the research to keep the seeds alive, but also developing useful information like what color and what size and all of that basic phenotyping work, and then distributing seeds to plant breeders. And so what I really want to make sure is understood is that this is one national system. There's many around the world, which I'll get to in a minute, but that national system, which is supported by the public, then is one of the major foundations for plant breeding through universities, through private industry, that then become the seeds that go out to farmers around the world. And without that system, it would be a much more fragile or unstable system going forward in terms of the future of new varieties and food. And so we even have backups of backups. This is probably the most famous seed bank or gene bank around the world, the Svalbard Global Seed Vault, which is our tertiary backup here in the United States. That is, we put seeds, we have our active sites, and then we put them in Colorado, and then we also put them in Svalbard, which is a Norwegian territory. For other countries, they don't have that Colorado version, and so their backup is at Svalbard. This is like a safety deposit box. You don't pay for it as a country, but you put your seeds up there and nobody else has access to them except you, so you can get them back when you need them. This is a picture of all the gene banks around the world, more or less, and the size of the circle is the size of the collections, just in terms of number of what are called accessions there. These are both national seed banks or gene banks, but also international ones, which are held in trust kind of for the global community, not just for a country. But I want to mention, even with the national ones like the USDA, they're not considered only for the country themselves. In fact, the distributions go out around the world. And so it really is it's a global network. And that makes sense because of that earlier map that you might remember, that crops came from places, but we need them everywhere. We grow them everywhere, the major crops at least. Maize is vital in Africa, and rice is vital in South America. For all of those reasons, we need genetic resources to be able to flow across the world. So how do you figure out if your collections are complete? If you need to go collect more, how are we doing in terms of making sure that these plants that are threatened in the wild because of habitat destruction, because of pollution, contamination, and overharvesting and all kinds of things, how do we make sure that they're in the seed banks? So there are techniques that are geographic to try to start to understand where the species live and whether we've gone to collect them. And that was a good part of my PhD, was starting to do research like this. This is basically the richness of beans, of Phaseolus, the whole group of 80 or so species. And that darker color in the middle is where they're richest in the world, which is central Mexico. And then the picture on the right is where they are richest, but we have not collected them well enough yet. And so that also became a major paper during that global project that was kind of all the wild relatives of all the major crops around the world, over 1,000 wild relatives that we consider the most important. And so this is an old figure. It's actually a little bit hard to see at this point, but the dark red would be the richest places to go for wild relatives if you wanted to find as many as you could in one single 5 kilometer by 5 kilometer grid. But let me just get a little bit more into the methodology that we use to try to explain it, what we're trying to do here on the conservation side or planning side. So this is just one of our wild grapes that occurs in Texas, Sweet Mountain grape. And what we do is we bring in information that come typically from plant museums called herbaria, or other types of museums, or increasingly from citizen science applications like iNaturalist, where people have marked, I've seen this in the wild and this is where I saw it, which you can now do on your phone very easily. and map those. So this is where the species has been seen, those gray spots. So it occurs basically in central Texas. And there's one other dot that you'll see there, which is the same kind of thing, except this is actually where it's been collected and put into a germplasm repository, a seed bank, or a botanic garden. And so you can already get a sense that, okay, well, it's been collected once from one spot in the farthest north. Is that enough? Do we have all the genetics that we need in the future? Maybe, maybe not. Okay, so now all this geographic stuff is based on hypotheses that geography has a relationship to genetic diversity. That is, if you go to somewhere different, then you'll get different genetic diversity. And so we do some modeling. So this is a guesstimate of where we think the species occurs based on where we've seen it, plus climate and soil attributes. And so the green is where we think the species might occur. And then we can draw a buffer around where we've already gone and collected and say, okay, maybe we don't need to go back there in the purple, but all of the orange, everything that's left of the distribution we haven't been to, is where we might want to go back to and collect more of. And that's what we call conservation gap analysis. And so actually 12.5% of the distribution, the posited distribution of the species, has been collected from, and the rest hasn't yet, in terms of this rather simple methodology. We have some other metrics that try to collect from every different ecoregion or different soil type and such, to try to get an idea of sort of the ecological variation. And then the other side of it is now not thinking so much about gene banks or seed banks, but thinking about how well protected are these species in the wild, that is in national parks, forests, and such. And so we have a similar methodology for in situ of essentially how much of that range is in national parks and such. So this was put together a few years ago into a national gap analysis that, that tells us how we're doing in terms of 600 of our native wild relatives here in the United States. And this map is a heat map, a richness map of all of them together, 600 species. And what I'd like, hopefully, to get two messages out of this. One is, no matter where you go, there's a wild relative underneath you. Even just these 600 most important to us in terms of relation to very important crops and such. The other thing is, up here in New York, maybe you're slightly out of that red, but you're in a hot spot pretty much here too. So there's a lot going on all over the place. The darker colors is more species occurring in the same areas there. Congratulations. You got work to do. So these are the kind of metrics that we came up with to try to understand how we're doing. And I mentioned a series of metrics around ex situ conservation status. That's in seed banks, gene banks, or botanic gardens. And then the other side is in protected areas. That's in situ conservation. And each of these black dots is one of the species, one of the 600. We have this scale from 0, which is we're doing very terrible on our conservation, and 100 is you're done. And that's why you can see green over there versus red. And you can kind of get a sense now. Ex situ, we got a lot of work to do. Most of the species are in that high urgent priority category for further collecting. In situ is a little bit better. If you combine them, we still got a lot of work to do. This is just grouping the species in slightly different ways into the types of crops or their associated crop itself. And I'll just take some water while you take a look at this. All right, so another contractual obligation of being a conservationist is, to have the doom and gloom story just a little bit. So there's my doom and gloom story. We got a lot to do to really conserve these resources. But again, let me just emphasize, if we don't have them in the seed banks and gene banks, they're really not that accessible for the plant breeders, right? And so we got to be doing that work. So how do we also make this information useful so people can go out and do that work? We've been building websites and making the information publicly available and all of that. So this is a really great website where you can actually explore those 600 species on a USDA ArcGIS basically site and try to get to know where they are and how well they've been collected. If you're a coder, we've developed an R code called gapanalysis.r where you can do your own analyses. And we're rebooting that because it's a little bit outdated right now with its packages. Very excited that we're now working on a project for the non-coders, which are most gene bank people, by the way, where online you can bring in data from some of these big global database sites like GBIF, which is the Global Biodiversity Information Facility, or iNaturalist, and then you can bring in your own data as well. You can figure out which of those points are just sightings versus which ones are actually in gene banks or botanic gardens. You can make your own buffer size, and you can do your own gap analysis. This is not quite yet ready for, you can go play with it already on that link, or there's a link on my last, on the last slide, but another year, and hopefully this will be really ready for prime time. I will say, unfortunately, this project was just canceled due to federal funding, but we're hoping to still move it forward. iNaturalist, I have fallen in love with citizen science myself. Just mentioning, I created this own group, a project on iNaturalist of just what are these wild relatives in the United States, and there are 2.7 million observations of observations of these species around the country. And so what an incredible resource. Even if 10% of them are misidentified, what an incredible resource. It really doesn't matter. The amount of information on them is just incredible. And I'm fully in on the citizen science thing. It's incredible. We've been working with our Forest Service and other land management partners to understand what's on their lands and to be able to protect them better. This is a really incredible story of how these collaborations started with land managers in the United States some 25, 30 years ago. We actually had a designation already of an area that was on Forest Service land, but hadn't been designated yet for anything around botanical interest, and it was designated as a wild chili botanical preserve. It's south of Tucson. If you're ever in that area, you can go see it. And so it's still special. Essentially, the US only has one or two really sites on public lands that are explicitly to conserve a crop wild relative. And this is one of them. Why is it there? Well, this is the progenitor of chili pepper, Capsicum annuum, and it just barely creeps up into the United States and a couple of places in Texas, Arizona. And this is conserving one of those populations, which is near Tucson. Very cool. So trying to expand that work by letting the Forest Service know what's on their lands. And so we're doing that kind of modeling for them on all Forest Service lands of which species might be there, and then going out. And a further version of that that then starts to integrate genetics, which is really fun, is a wild cranberry project, which is a collaboration between USDA's Ag Research Service and Forest Service, and identifying, in this case, actually across most of the East Coast, different populations of wild cranberries, and then doing genetics to figure out how much they differ genetically to identify kind of the most interestingly different populations that might be specifically designated in these categories. And so you can see just by the vegetation here how different the ecologies are. This is everything from, well, actually there's some West Coast stuff here, but everything from kind of north all the way south to West Virginia. And so pretty cool stuff. This presentation is certainly not just my research. It's also colleagues around the continent, around the world that are doing work. And so just a little bit on the Canadian and Mexican efforts here. There has been, as mentioned, quite a lot of just inventorying and then mapping and trying to understand where the species are. And so as thought and as expected along the southern border of Canada is where you get the richest. And that's also true to some degree in the United States, although as you saw, there is this northern range because of so many berries and things that are more temperate there. But we have a lot of richness also on our southern border in Texas, Arizona, New Mexico, Florida. A lot of berries up there in Canada that are, I think, a lot of the efforts of the people who are most active right now doing work in Canada on the native wild relatives are around their cranberries, which are culturally significant, also significant for wildlife, et cetera. Mexico, huge diversity, also a lot of studies of who they are and what their risks are in terms of disappearance. I want to convey a message that it's been driven home to me by colleagues from Mexico, which is somewhat of a different way of framing and understanding these species, because there is still a lot of integration with traditional agriculture, with the wild relatives. And so there are systems still with beans and maize and other cultivation systems where the wild relatives are occurring in the same area as the domesticated fields, and there is gene flow, often both ways. And so they're understood in less as just sort of this kind of historic thing that you can bring genes and take them and put them in, but also as part of this dynamic system of traditional agriculture where there is still gene flow. And that is a different way of seeing it and an important aspect there. We've been doing work together, publishing books and such around what is this richness and what are we doing about it. A couple of volumes, if you're interested, in over 1,000 pages of North American crop wild relatives. And then starting to really reach out to a wider audience, including the public and eaters, diners, if you will, around these wild relatives or wild foods. And so this is an example of several different events that we've done. This one was in Tucson, where it's a wonky, I think it's a funny experiment in and of itself. Can you kind of do wonky sciencey stuff, bring together scientists and talk about these things? Can you also throw parties for the public with chefs that bring in the public that want to taste alcohols made from wild relatives or interesting food? Can you go down to preserves like the wild chili preserve and see them? And it's a interesting experiment. I think it's been very fun to try to message this still kind of middle land between wildness and agriculture, threats and food security all together into a message that kind of combines all of our challenges in the world and all of our issues we got to work on. I've been working in the Botanic garden world for the last 4 years, and so I want to mention that because it's the other side of the whole world of ex situ conservation. That is where we put things into frozen storage or into live plants in gardens. And just as you saw that picture of all the gene banks around the world, there is also these thousands of botanic gardens around the world who are also holding crops actually and their wild relatives. And they do it for somewhat different purposes, but we're making efforts to come together to do conservation all together, all of our repositories. And so one of the ways for arguing for that, which is a little simple, but if you'll allow me, this is another map of the richness of wild relatives in this area, but those buildings are the different USDA sites that I mentioned, the active research and collection sites. Colorado's there in the middle there. So you can imagine the number of different people working there and what they're able to do, but then you map all the botanic gardens on top of that and all their botanical expertise around being able to identify species or go out or collect and all of that. And you might imagine that you could do more working together. And so I got to be part of a really fun project almost 10 years ago that Dr. Mark Sorrell also up there was part of. And we had never met each other actually before, and it was really, really cool. We were both on the sort of the more ag side. There was all these botanic garden people that I'd never met, and it was all about like, could we work together and could we do something on crop wild relatives? And so we decided through that to come up with a roadmap for North America on what do we got to do. And we published that in the crop journal. We published it in a botanic garden journal. We published it for land managers. And it has 5 tenets to it. This is kind of the big picture of how do we deal with this giant richness of diversity that is threatened, that is important for our food security, that plant breeders need. And so the first one is we still got to do science about who are these species and where do they live and how well collected they are. The second one is we got to protect them in their natural habitats. Why? If they're not still evolving in their natural habitats, then they're not evolving alongside pests and diseases and changes in the weather and climate and all of those things. The third is we got to put them in the seed banks and gene banks so that they're conserved for the long term, but they're also available to plant breeders and others. The fourth is we got to do the value addition of phenotyping and all of that stuff, pre-breeding to get them ready. And then the fifth one is public awareness and outreach, which the botanic gardens are particularly posed to do good work on. And so just a couple kind of last slides that are about that networking or that growing network of people that are really working on it. So as the Botanic Garden world then and the gene bank world has come together, we tried to figure out what is the low-hanging fruit for us to work on. And it actually turns out that fruit and nuts is a low-hanging fruit for us because gardens have live trees. They often have whole plants and have enough space for them, whereas the gene banks struggle to have enough field space for them. And so it makes a lot of sense to work together in that way. There's gardens that have giant apple collections, for instance, the New York Botanical Garden in the Bronx. And so this is a network that's been happening for a couple of years. It's working on these 10 genera. If you're interested in any of them, please get in touch with me. And it's a very active network of understanding who the species are. It's got a little bit of money so that folks can go out and do collecting and then bring them into the botanic gardens or gene banks. And this is 3 pictures from my own work at the San Diego Botanic Garden, bringing in these collections. This is 2 wild plums from native to California, one of which is very, very rare and endangered, the desert one on the left, and then a wild walnut in the middle. It's really fun. It's a great network of people. We also put on a symposium last year that was quite an experiment, I would say, as well. It's kind of a reboot after that first work 10 years ago of getting together, and it was where we at and how do we go forward. And there's still some really good resources on that website that you'll be able to find there. But yeah, before I get to that, let me just go back here. So one thing that I really hope to express is that I think also in the last 20 years, as our working in this field has deepened in this region, we've also started to move beyond that simpler understanding at first that all we're doing really is just grabbing genes and bringing them over, which is wonderful and important. There's nothing at all wrong with that. But the bigger picture of what are these wild plants and how can they be used? And that has broken down all kinds of terminological silos, like wild or even crop. Let me just repeat that. So like crop, first of all, what is a crop? Does it have to be cultivated? So what about the plants here in North America that aren't particularly cultivated, but are food resources and very interesting, like pawpaws, or a plant I'm about to show you in a minute? And then what the heck does wild mean anyway, as we start to realize that people have been managing wild populations forever anyway, through burning or selecting or management. And so our understanding is less now that there's just wild stuff and you bring it over into the domesticate, but there's this giant spectrum of plants that have been associated to more or less of a degree with people for thousands of years, and we're still in there somewhere. And what do we want to do about that, and where do we want to go forward from here? And I'll get back to that in a minute for a last point, but on the networking side, I wanted to mention something actually very exciting, and that's that in the Botanic Garden world, they've started to have these global networks to do important work on really important taxonomic groups like oaks or cycads. There's a cycad out in that nice little green area out there. And these are often very threatened plants. And it's this whole group of thousands of institutions through a network like this getting together to conserve all the species. And I'm very excited and proud to say that the 12th, quick math there. The 12th Global Conservation Consortium has just been agreed upon for the world, and it's going to be focused on food plants, including crop relatives. And so that really brings the whole Botanic Garden world of thousands of institutions connected with gene banks around the world to try to do something about crop relatives, but also about all these wild food plants that are increasingly threatened around the world, et cetera. And so, okay, so now finally I get to this expansion. I have 3 more slides, and I think I have enough time to express them. I'm going to take a deep breath for a minute and try to give you one last big thought. This is such a great institution, and so many amazing brains and creativity here, so much productivity. I really agree with the previous speakers that spoke about what an amazing place this is, and I'm really honored to be here. Taking care of our major crops, I think in the title of this symposium is Wild Relatives of Our Major Crops, is of utmost importance in terms of those crops' future. We have to keep this diversification process going on or bringing in new genetics. That said, my own feeling is that that's just the beginning of what agriculture can look like with these wild plants or semi-wild plants. Don't even use the word wild, these plants. And so, anybody know what this plant is by chance? Please. It is a groundnut, American groundnut. So this is Apios americana. This is a plant that that was incredibly important in Native American food systems. You could say agriculture if you'd like, all the way from St. Louis to Cape Cod at one point, and is kind of barely around anymore, mostly by hobbyists. There is a USDA collection in Iowa. This is known to be really associated with people because it's a triploid. It has a native distribution as a diploid, a fertile diploid, but these triploids are what were occurring all the way from St. Louis to Cape Cod with bigger tubers. It's a legume tuber, higher protein. It's tasty. This is my own actually harvest in Colorado, which is kind of marginal to collect it there, which I fed during Thanksgiving to my family. It was known to be probably one of those foods eaten in the first Thanksgivings. And of course, my family had no idea how there was like teaspoon each that they got to eat. But I thought it was pretty darn good. It's an amazing plant. And it's just one of these plants that I want to express for those that have the opportunity and privilege to think outside the box about new crops. This is one of these crops that really could be something. And there's an association in the world with tuber people who are predominantly dependent on tubers and protein deficiency. And so a crop like this could also be an incredible crop for other regions in the world. Just wanted to mention two other examples that I think are really neat that are coming along that are not so much about bringing genes over, but thinking about bringing entire plants into food systems or our agricultural systems. And this is work by my colleague, Dr. Nolan Kane, and colleagues in University of Colorado, also USDA and other places. And this is a native flax that you can see in its native distribution in that map on that side, widely distributed. It's a perennial. And so this is work that was also referred to a little bit in an earlier presentation around the promise or opportunities of perennial crops and in more sustainable agricultural systems. This is a pretty neat one. It already has good oil content, like the domesticated flax that is mostly grown, and it's already in wildflower mixes, so we kind of know how to grow it already. It's really just figuring out the agronomic systems and bumping up the quality and the quality essentially. And so there is some really great work like this going on, and I'm happy to talk more about it to those who want to talk more about that stuff. And so as a last slide, so here's a question that my mentor and my postdoc, Dr. Allison Miller from the Danforth Center in St. Louis University in St. Louis, asks, which are the wild relatives of our future crops? And her argument, which I agree, is they are certainly the wild relatives of wheat and rice and maize and tomato and brassica, the canola and all of that. But if our future crops are also other things, then our wild relatives are also other things too. They can go much beyond. And so there is some really excellent and interesting work about trying to figure out, for instance, if you want to go perennial, how many herbaceous perennials, in this case, the Land Institute type thinking on productive perennial systems that you could mow and things like that, how many are there out there in the world? And so in terms of grasses, in fact, there's almost 8,000 perennial herbaceous grasses that could be looked at. And then how do we whittle that down to 20 or 30 or 50 or a couple hundred? that might really be promising next step crops for the future. I'm so inspired by this because this is the kind of long-term, big picture thinking that maybe 500 years from now, we end up with 20 or 30 or a couple hundred different crops that we're eating, and how exciting and interesting that would be. So I hope this was interesting, and I hope that it just drove home just a little bit how important the foundational bigger thinking and conservation work is to plant breeding. Lots of links to these websites and data resources and papers at the end over the years. So thank you so much. Happy to answer questions. Yes.
Thank you so much for the presentation. I was wondering if you have any knowledge of or interest in crops being domesticated by non-human animals.
So, and how, if you don't mind me asking, how do you define a crop in that case then?
Ooh, oh no, I know. That's a good question, because I think I'd probably put human in the definition.
Yeah.
Or say that it's a plant that's been used widely by humans and has diverged from a wild relative, but I guess you could say the same.
Yeah, well, I'll try to answer your question in some helpful or meaningful way in any case. Well, I don't have off the top of my head something that, yeah, I do actually. I have two answers.
All right.
One thing is humans are incredibly innovative. I mean, obviously, we're doing all kinds of crazy things like cell phones, right? I still think probably the biggest, most incredible innovation that's ever happened is agriculture itself, which is this huge process of just of noticing what's around and then working with that diversity and changing it in this co-evolutionary process where everybody needs everybody. Humans need the plants or animals, et cetera. But it wasn't just us. And in fact, we capitalized on other animals quite a lot in some cases, particularly with fruit. And so avocados, for instance, didn't actually go from this to that like maize did. Avocados actually went from this to that because they were already being eaten by and selected by megafauna in this case in the past. And so I think maybe that's something that gets to your question. But the other thing I want to say that's also really important is, as many of you probably know, the more we know about plant systems, especially plant systems, and what we're hoping is more sustainable agricultural systems that have living soils and such, the more we realize that the cropping systems are actually very complex with microbiota, with pollinators, including things like bats for agaves and things like that. And if those things aren't all still working, then you really don't have your plant system that in cases like agaves are sometimes a little bit contentiously, I know, but not even called plants anymore, but holobionts. That means that there's so many organisms that have to work together that if you just have the plant, it's really not the thing anymore. It's not the crop. You need all the microorganisms, and you need the pollinators, and you need the people, the whole shebang.
Cool, thank you so much.
You're welcome. Yes.
So in my mind, and I haven't researched this recently, so I could be wrong, but I feel like a lot of the threats to conservation and natural diversity are driven by capitalist market forces. And I always thought that it's kind of hard to quantify in the language those forces are conversant in, like economics, like the value of biodiversity. If people are really like deeply steeped in plants and agriculture and wilderness, like that value is kind of inherent maybe in our perspective. But now that you seem to have this very high level picture of genetic diversity in these crop wild species, like across America and kind of across the world. And we're also developing a pretty clear understanding of the impact that diversity can have on our agricultural system. Do you feel like there could be kind of a quantification of the potential economic value of genetic diversity in these crop wild species? And would that quantification be a useful argument for conservation efforts if it could be made?
Cool, great. I know that there have been for 30 years economic valuations of genetic resources, including crop wild relatives. And among research groups, there's a person named Melinda Smale, who I think had some associations with Cornell, in fact, and some in other places. And she did some preliminary work as well as a group in USDA, the Economic Research Service. So you can look up some of that early stuff. It's pretty wild. In some cases, the valuations are specific to genetic innovations, and they're always in the millions, if not billions of dollars. And so if the economic arguments, I think, have been there. To do one for the whole, like what is the value of wild relatives economically, I don't think anybody's come up with the single number. And of course, it's a crazy thing to even attempt, but if there are any economists in the crowd that want to give it a try, it'd be incredible to hear that. I don't know, I'm not placed to say as an economist or policy person or anything, which I'm none of those, how effective those arguments are, but I do think that money talks to some degree, and we should still be speaking in those languages to drive home the ever important message that we need genetic resources. Thank you.
Sorry. I think we need to move on to the next speaker time-wise, but yeah, definitely find Colin.
Thanks so much.
Thank you, Henry, for the kind introduction. I want to thank the symposium organizing committee for all their hard work in putting this event together, as well as to Corteva for sponsoring the event. I'm really excited to be here today to share some pieces of my PhD research with you, which is focused on maintaining diversity within the Kansas State University wheat breeding program. So when we think about wheat performance over time, we have seen sustained genetic gain in terms of wheat yields, specifically around the adoption of Green Revolution varieties in the 1970s and 1980s. We see a significant spike in our wheat yield increases. However, this genetic gain for yield, while is still increasing, is decelerating to where this might suggest a potential plateau and improvement in wheat yields. And at any point when we start to lose improvement for a certain trait, it can become very prudent to go back and look at those more diverse sources or wild relatives to be used in breeding. When thinking about what makes up the overall wheat yield, we can think of it as a trade-off between our source traits and our sink traits. So our source traits are going to be those traits that are more important in the early stage of the growing season. Development of adequate levels of biomass will determine photosynthetic efficiency and eventual translocation to the sink filling organs, which are on the right side of the triangle. The sink filling organs are going to be more important during senescence and grain filling period, to where these are the ones that have been historically prioritized the most in increasing wheat yields. Most of our increases in wheat yield over time has come because of increases to harvest index, which is shown in this bipot analysis, where grain yield and harvest index have the highest amount of covariation compared to these other yield components. So in thinking that many of our improvements to yield have come because of harvest index, if we want to keep making improvements, we may need to start focusing more on this right side of the triangle in those source traits, while keeping in the back of our minds that we have to make sure to optimize this trade-off. You have to have enough biomass to photosynthesize early in the season, but you also have to leave enough time to fill grain later on in the growing season. So in thinking about how we can do this in wheat specifically, wheat has a complicated genetic history in terms of its evolution and domestication, which provides several opportunities for crop wild relatives. I was very thankful to Colin for introducing the onion concept of the primary, secondary, and tertiary gene pools. When thinking about wheat, the primary and secondary species that are in the gene pool are going to be those ones that have genomes in common. So those are going to be the ones that are shown on this slide. So to go over just in detail the steps that occurred in generating our hexaploid wheat or Triticum aestivum. The first species that were involved were the wild grass species, which donated the A and B genomes to form tetraploid Triticum turgidum, subspecies dicoccoides, which is also known as wild emmer. Further domestication made cultivated emmer or Triticum turgidum subspecies dicoccum come about. After the free threshing character was conferred, durum wheat arose. This is what is used in our pasta production. These free threshing tetraploids were also the tetraploid donor for Bread wheat, which when hybridized with the D genome donor Aegilops cowshii, formed hexaploid Triticum. Some things to notice about the evolutionary history of wheat is that there were 2 hybridization events, each associated with their own genetic bottleneck. Because of the makeup of the wheat genome, there have been many opportunities to lose out on genetic diversity coming from these wild relatives. The other thing I want to point out is that this species, wild emmer, specifically can be considered the central conduit of gene flow between the wild diploid species and our cultivated durum and bread wheats. So there are several main approaches to gene introgression. A very straightforward approach would just be through making direct crosses. This will be limited to species within the primary and secondary gene pools, to where meiotic recombination will not be very successful if the chromosomes can't even pair in the first place. Synthetic wheat is another approach that's taken specifically in wheat breeding. This is where a cross between a tetraploid durum or other tetraploid wheat is made to that D genome donor. This basically regenerates that initial cross that resulted in hexaploid wheat. However, a drawback of using this approach is that you're essentially discarding many generations of breeding progress that has been made in hexaploid wheat. So these materials aren't generally very adopted agronomically. Gene editing also has its place in introgression breeding. This is really specific for precise edits. When thinking about just increasing overall quantitative variation, it can be a little more challenging, and it's also important to think about any sort of regulatory constraints that come along with that process, especially in the US and in working with wheat germplasm. Amphiploid hybrids are a really interesting approach that can be taken. This allows for crossing between 2 relatively unrelated species, because when that cross is done, artificial chromosome doubling is performed. That way, the complete chromosomal constitution of each of the 2 species that were used in that cross is maintained. So the amphiploid hybrid approach was what was used in development of my PhD research population. So I know this is a lot, but just go step by step with me. The first step was through crossing the hexaploid recurrent parent bread wheat donor to the Aegilops cowshii D genome donor. A cross between these 2 species does not have much match in terms of its chromosomes, which is why we had to double the chromosomes with colchicine. This resulted in development of a synthetic octoploid, where we have the full genomic makeup of Activum and the full genomic makeup of Aegilops cowshii. In this way, this synthetic octoploid is used as an amphiploid bridge to bring in the diversity coming from wild emmer or Triticum dicoccoides. So that second step was using this amphiploid bridge with a cross to dicoccoides. After that, the hexaploids are able to be regained. Something that's important to notice is that in this F1 generation, we still have a really high contribution of theoretical wild alleles in our background. So to regain that background recurrent parent, 2 back crosses were made to the same hexaploid recurrent parent, to where after the first back cross, the theoretical proportion of wild alleles would be around 30%. After the second, that's reduced down to around 15%. So essentially, this population is developed to have a very high proportion of that recurrent parent background in it to maintain adequate levels of yield and regional adaptation, while still keeping low-level contributions from those wild relatives. But it's also important that we keep those introgressions at a high enough frequency to where we can actually pick up on any meaningful variation. So after these F4-derived F5 lines were increased, we were able to do a ton of phenotypic trialing across Kansas. We used 3 years, 3 locations distributed across the precipitation gradient. However, it's interesting that in the 3 years that we did this study in, all of Kansas was under a pretty severe drought. So the differences that we might observe between these locations in a year with adequate precipitation were not necessarily observed in my data. The largest piece of my project was collecting this massive quantity of phenotypic data. I'll go over each of these components in the next several slides, but the goal was essentially to look at the trait means and variances of those wild introgression lines compared to common check varieties that are used as benchmarks of regional performance. So before I talk more about each of those trait groups, I wanted to discuss my data processing. So this is essentially a zoomed in view on 2 of those 279 introgression lines. What is shown here are my raw observational data, Y, for 3 traits: senescent state, plant height, and grain yield. What you might notice is that these traits have very different magnitudes of variance, to where making efficient comparisons between them can be really challenging, especially when considering deviation between them. So that's why I pursued a data standardization approach. This is a fairly straightforward methodology to where within each location, I took the within environment trait mean, subtracted it from the raw data, and divided by the within environment standard deviation. After doing this standardization process, we're left with a data set that is quite more easily interpretable, to where the mean is going to be at 0, which means anything with a positive value is going to be above the mean, anything with a negative value will be below the mean. This just becomes a really easy way to view a lot of data at once. These standardized data were used as the input for 2-stage analysis. The first stage was modeling the within location genotypic values as fixed effects. And we did this by making a AR1 row column spatial correction. After running this model, we had our blues, or genotypic estimates, and our weights corresponding to each one of those values, which were used as the input into the next model in the 2-stage analysis, which is a BLUP model. This is run across locations using genotype as a random effect, to where we can extract one single genotypic value for each genotype across all locations. Doing this enables much more efficient analysis, because instead of doing single within environment analyses and trying to composite across them, this essentially does that for me. And to further verify that this approach was a reasonable one, I looked at the genetic correlations of different traits and traits within environments. So the diagonal shown here, or the matrix shown here, has genetic correlation of grain yield blues at the 6 locations tested, with genetic repeatability on the diagonal. What can be noticed is that the high degree of correlation between these locations indicates that there aren't very many rank changes in these genotypes, which allows me to make more efficient comparisons just using those BLUPs. I would expect my results to apply regardless of my location. Looking at my trait BLUPs in this matrix, again with the genetic repeatabilities plotted on the diagonal, I specifically want to highlight the fact that these repeatability values are moderately high to high, which is not necessarily always expected for highly quantitative traits, which is what I'm working with. But the fact that those had high repeatability values indicates that using these BLUPs is going to be an effective way to still capture that effective variation while not having to necessarily do it within each environment. So going into some of these specific trait groups, one of the first things I measured were some of my phenological characteristics, so heading date, senescent state, grain fill period, and plant height. A pattern that is interesting here to notice is the divergence of the mean and standard deviation of the checks versus the introgression lines. So we have this orange bar corresponding to the checks, and the purple corresponding to the introgression lines, with the mean centered at 0. And we see that these checks are on average developing quite a bit later compared to the introgression lines. And this can be localized to a region on chromosome 2B. And I apologize, I didn't have time to talk too much about my marker data, but I did want to show some of my GWAS results just so you guys can see that we are still able to pick up variation coming from these wild alleles, even though they're at very low minor allele frequencies, which is shown in this table here. Moving on to yield components, something that is important when thinking about working with wild material is the fact that we're going to see pretty significant yield drag. So these are showing grain yield for the checks, for the introgression lines. We see that those checks are having a much higher level of yield compared to those introgression lines, although that pattern doesn't necessarily follow off of throughout all of our different traits. So focusing first on our traits that are related to the source stage of the yield triangle, we saw quite a bit of variation to where I got some fairly strong peaks across all of these traits. And a specific thing I want to point out here is that for my green reflectance values, I got that same peak on chromosome 2b. This is fairly intuitive, however, because previously when that peak was associated with maturity and development, to where something that is senescing is losing its green chlorophyll content. And so when you average that green reflectance throughout the growing season, things with lower values are probably going to be the ones that matured faster anyway. So far, making sense still. Moving on to the sync-related traits. We see that there was a common association for several of these grain size traits. So 1,000 kernel weight, test weight, and kernel width. Specifically, that association for test weight, which is essentially how effectively your grain can pack into a certain volume. That has a fairly large effect and fairly large percent variance explained, to where the fact that this region came up from multiple grain size traits indicates that this may have a fairly large effect on the yield drag that is observed in these materials. While we see a drag for yield, the opposite effect is generally observed for our quality traits. One of the reasons people use wild emmer specifically in wheat breeding programs is because of its quality. When thinking about our grain protein content, we see that these checks have an average much lower compared to the wild introgression lines. However, grain protein and grain yield in wheat have a very strongly negative relationship, to where it can be challenging to select on both of these things. And if you're going to pick one, then you're going to pick yield. So this is where breeders use a metric called grain protein deviation, which is essentially a regression of protein content on grain yield, using the residuals from that regression as the protein deviation values. This can be used in a way to where a cutoff threshold for grain yield can be established, and then lines with higher than average grain protein can be selected, which is shown in this green box. And so looking at the distribution of grain protein deviation in my population, which essentially combines the protein and yield, we see that those checks and their current parent all kind of cluster at the lower end of that distribution. So these introgression lines have higher grain protein deviation overall. But one thing that is interesting to notice is that for protein quality score and solvent retention capacity, which while grain protein is more a measure of protein content, these are a measure of protein quality. Those do not necessarily increase. So while we've brought an increase in protein content over from these wild relative introgressions, we did not necessarily increase the functional protein quality. So I also screened some of these through a full ionomics panel of 19 elements. I didn't include these on the previous correlation matrix just because it would have been incredibly massive to look at. But I showed these here, also including grain yield and grain protein. Something to point out is the generally negative correlations between grain yield and these ionomics traits, meaning that selection for increasing concentration of desirable elements in the grain may very likely reduce our grain yield levels. And additionally, when looking at these genetic repeatabilities, those low genetic repeatabilities indicate that there's a lot of environmental variation that's going to have an effect. And so the efficiency of selection won't be that high anyway. So these ionomics data are a little bit messy in terms of how we can use them for breeding. But nonetheless, I was able to actually find an interesting pathway which can be pursued, to where for a rubidium, which is shown here, I got that same peak on the same region of chromosome 2b. So rubidium is generally used as a proxy for potassium transport, and which is going to be water transport. So when you see variations in your rubidium concentrations that are localized to the same regions as your development, that can really be indicative of some sort of a drought-related effect, especially given the fact that these data were all collected under drought conditions. So it's going to be interesting to look more at this region on chromosome 2b in greater detail under more controlled drought environments instead of just the randomly natural drought that my experiment was conducted under. So I've shown you guys a bunch of data. Sometimes my introgression lines did better, sometimes my checks did better. a question that comes up is how on earth are we going to use all of this data? So this is where index selection becomes really interesting. And because I am in an applied breeding program, I'm still going to always be the most interested in grain yield, which is why I developed this selection index specifically to always pick the highest yielding things. So the way I did this, and just a reminder that these are standardized BLUPs I'm working with, I multiplied the BLUP of grain yield for a specific genotype by the BLUP of another trait, so in this case, grain fill period. I selected a subset of traits that I thought captured what breeders are generally selecting on, so grain fill period, high yielding but low short plants. That's why I divided by plant height here. I had my protein, my solvent retention capacity, some of my kernel traits. And after doing these individual grain yield by trait indices, I was able to average across them to come up with an overall index for all of these lines. This index pulled out a couple introgression lines that outperformed absolutely everything in the population, including the highest performing check, which is bolded to showdown. And I have this plotted next to grain yield in bushels per acre for comparison. And you can see that while the selection index is pulling out the things that are best across a wide range of traits, it is specifically also pulling out the things that are the highest yielding, which is going to be incredibly important when thinking about developing breeding materials. We weren't necessarily trying to develop germplasm to release. We were more thinking about developing parents for our crossing block, which these materials are already being recycled in our crossing block this year. And additionally, these top three, so those three are being pursued for germplasm release later this year. So this was a really positive result out of my project because we weren't necessarily expecting that yield and adaptation to come through from that recurrent parent. So overall, some of the most interesting things that I have found out of my project is that using that amphiploid hybrid bridge approach was pretty effective at bringing in diversity on all three genomes at a level that was able to be detected by my different analyses while still maintaining that recurrent parent background. These things are still very well adapted to Kansas, even in spite of the drought conditions that was experienced. Additionally, there are several interesting routes that can be pursued further. I think that region on chromosome 2B could be really studied in a lot of detail. Also, the region on 6B, I think that would be really interesting to look at in terms of reducing that linkage drag that comes from wild introgressions. So with that, I just want to make sure that nobody thinks that I did this all on my own. I have to give the biggest of thanks to the K-State Wheat Breeding Program. They've supported me through every single stage of my research. Additionally, my graduate committee for all their intellectual contributions. I was pretty stupid when I started, so they've helped me become slightly smarter. Also, my mentors, they've been the people who have gone out of their way to make sure that I have had the one-on-one support I've needed at any given point in my research. And of course, my family and my funding sources. So with that, not sure how much time we have left for questions, but you can find me during coffee, and thank you. Yeah, you can go first.
So you talked about how you're developing these recombinant and red limes, and you had a certain estimated proportion from each of your parental species. And that's assumed in billions.
Yes. However,
did you see any segregation distortion?
That is a good question. So I did check for aneuploids, did not find any because since these are BC2s, that allows for a lot of that background to be brought back in to where these were mostly the recurrent parent. I just got access to the deep sequencing data from my parents. So that's the last big piece of my project. And so I'll be doing introgression mapping to like more finely delineate where those introgression regions occur so I can look more at that segregation distortion. I also expect those frequencies, once I do that, to not necessarily match my Mendelian expectations. Mendelian expectations do not necessarily apply when playing around with ploidy levels super well. So these are approximate estimations. Additionally, there was some negative selection that we did against things that didn't thresh or that shattered before harvest. So I might expect those two have a higher proportion of wild alleles in them, which is why they were not adapted. And removing those would subsequently lower my overall allele frequencies from those introgressions. But if they can't run through the combine, then there's no point in evaluating them. Thank you. Any other, oh.
I was just curious, you mentioned the overall protein content and then the protein quality. And I was curious what that meant, like quality in terms of nutrition, or what were you looking?
Yeah, so the question was about protein content versus protein quality. So we have a really breeders friendly low or high throughput assay for early generation materials because it only requires 5 grams of seed, and it's a lactic acid SDS SRC. So we basically add lactic acid and SDS, and we can do the dry pellet weight, wet pellet weight, and adjust that for moisture to look at our solvent retention capacity, which gets at your gluten strength. These are very well correlated to some of our lower throughput bake analyses that are done on our very late generation materials. And then off the solvent retention capacity, if you divide that value by the protein content, that gives you an estimate of protein quality, because something that has higher solvent retention capacity but lower protein has more functional protein. If that makes sense. So yeah, it's a breeder's assay that was specifically developed for wheat breeders. Thanks. Yeah.
Could you elaborate more on the ionomics section? And you said you're using the rubium levels to infer drought resistance, is that?
Mm-hmm. So rubidium has a pretty high or strong correlation to potassium, which when you look at the potassium concentration in the grain, that's going to be an indication of water movement throughout the plant. And rubidium is used as a proxy for potassium transport because the way that they are seen in these assays is relatively similar. And I had to learn that from some little deep dive in the literature. Did not necessarily know that. And so when we see things with elevated rubidium levels, that can indicate that the plant is under higher levels of drought stress, because if there's more rubidium, that means there's going to be more water transport. And so that's kind of where we're going with this association right now. This will, of course, be better verified with my deep sequencing data and my and any other analyses that are done on this region. I don't know if that answered your question. Perfect. I think I saw a hand over here. Oh, yeah.
So this is a bit more anecdotal and like maybe far out of the distribution of your research, but I noticed in some of my work that I look at chloroplasts, and the chloroplast genome for a lot of Aegilops species, and some Triticum is like drastically shorter than others. So I was curious, when you were doing your crossing, did you like observe anything weird happening based on cytoplasmic inheritance? Like if you had Aegilops as the maternal, the female parent, the cross versus not?
Yeah, so that's a good point, is that when crossing with these wild relatives, it can be really hard to use the wild relative as a female parent. We used the wild relative as the pollen donor in all of our cases, because especially with like Aegilops tausii, those are some really small anthers. It's going to be hard to pull those out. So yeah, we did use the recurrent, whatever had the recurrent parent in it, that was always our female donor. So we didn't specifically look at maternal versus paternal inheritance, but that would definitely be interesting to do.
Yeah. I wanted to ask, what has been your greatest challenge in this research, and also what is the thing that you're most excited or passionate about?
Most excited about is this. We were really not expecting these things to look very good in the field. They are not super drought tolerant. I mentioned that drought escape strategy. That's not necessarily desirable because that's going to shorten the grain filling period. So the fact that we were able to get upwards of 80 bushels an acre grain yield off of these materials was a really encouraging result, especially because there's simultaneous research that's going on alongside at our at the USDA in Manhattan, Kansas, using emmer in direct crosses, to where when making comparisons between things using the direct crossing approach and the amphiploid bridge approach, there's a significant difference, to where we really like the introgression strategy we used. And I would say the most challenging thing, I think that there are not a wide variety of resources to train graduate students in breeding analyses. if that makes sense. So the kind of some of the technical aspects, it's hard to get exposure to both the practical and the theoretical. And so being able to bring those two things together and kind of think at every stage of the process, what might be going on, and thinking of a plant as a whole system and not just puzzle pieces, but a whole all together. But I could list a million things.
Cool.
Yeah.
Thank
Thank
you so
you.
much.
We have time for one more question, but if not, then we will come back here at 3:25 for Samantha's talk. But enjoy the coffee, everybody, and give it up for Addy.
Potentially speciation. And so we're thinking about these processes happening at an evolutionary timescale. So we're thinking hundreds to millions of generations. And we have these kind of three main processes: mutation, selection, and drift, that are really big shapers of this genetic diversity. But sometimes there can be these hybridization events between two species, where instead of getting half of a genome's worth of DNA from each parent, you get the full genome from mom and the full genome from dad. So instead of 1 and 1 making 2, you get 2 and 2 making 4, or a tetraploid. And over these evolutionary timescales, we can see this DNA sequences are being lost. And this process is called diploidization, where that polyploid species becomes something like a diploid again. And this process of sequence loss is called fractionation. And while DNA sequence loss is a major consequence of polyploidy, and basically the response to this event, there can be other changes, including changes to gene expression or changes to existing gene functions to change slightly that gene function, or even develop novel functions. And it's not just the genes that are duplicating in these events. These are affecting all of the other genomic elements, including repeat space, like transposable elements, and also all of those regulators that are affecting gene expression. And so when we have these whole genome duplication events, we are generating this massive pool of genetic diversity, and thus a huge pool of adaptive potential. And this process of fractionation whittles that pool down. But during this process, we can also see these bursts of diversification, where there are many more forms of species, either forms of the same species or even whole new species forming, that could be adapted to different environments compared to that parental species. And so in this way, polyploidy and fractionation are major shapers of genomic diversity and structure, potentially leading to these novel forms. Okay, but you're probably asking yourself, well, Sam, how common is this actually? This seems like this would be a pretty rare occurrence. But actually, polyploidy is very common, particularly in plants. And so here, this is a wonderful kind of mini review about it, where they made a phylogeny using all of the plant emojis available to you on your emoji keyboard. And they put all of the whole genome duplication events that we have observed as red dots, and all of the whole genome triplication. So instead of being 2, right, you have 3, onto this phylogeny. And hopefully you can see that there's quite a large number of them. Now, if we have an estimate of what that genome size was right here at the base of this phylogeny, right? Either how many chromosomes was in that original genome, or how much DNA, as like in picograms, there was. We can then say, okay, if you've been through so many rounds of duplication, we can multiply that starting number of chromosomes and DNA weight to get an estimate of how big that genome should be now. And that's what's indicated here in yellow, with being chromosome numbers, and blue being that genome size and just total DNA mass. And with the darker colors being bigger genomes. But what do we actually see? We see much smaller genomes and much fewer chromosome numbers than we would expect here in those kind of inner circles that are much lighter in color. Now, personally, I find these color scales really difficult to parse and compare. And so I'd rather show you this histogram instead. Here we have genome size, right, in picograms on this x-axis for thousands of angiosperms. And the high edge of this histogram, while there is a long tail at much larger genome sizes, the edge of this histogram is at 30 picograms, which is about where the arrow is pointing to on the original scale. So as you can see, we're not even getting into those darker colors. It's all these basically gradations of bright, bright blue. Okay. So all of these small genomes have to be due to these really large amounts of DNA sequence lost over time, given all of these polyploidy events. And since in this talk, I really want to focus in on this process of fractionation, I want to reiterate this with a simplified cartoon. And so here, this cartoon is really nice in that we have a red genome and a blue genome that come together to form a polyploid that has 6 chromosomes, 3 from blue, 3 from red. And over time, we see sequence loss where maybe those chromosomes start to shrink in size. And maybe over some more time, some of those chromosomes start to fuse together. And so we see a reduction in overall chromosome number. And maybe over really, really large timescales, we see something that looks like a diploid or has been rediploidized, and we're back down to a chromosome number of 3. Now, this is a really nice, simple cartoon. But as a biologist, when I'm coming up to this species at the kind of end of this cycle, I don't see red and blue. I just see something that looks like a diploid with 3 chromosomes. And so the only way we can see this history of duplication, to parse apart those red and blue chromosome segments, is by aligning these quote unquote purple chromosomes to either the red or the blue genome, or something that's very similar to one of those. Okay, but what do I mean by an alignment? So here we have this ancestral species, and now each of these colored triangles represents a gene. And given some time, we might see speciation, giving us these 2 descendant species. Genes that arise from that same common ancestor, so the red triangles here, we call these orthologs. And when we see all of these orthologs kind of conserved in the same order and orientation, we call that synteny, right? This conserved gene order. Now, over time, structural variants such as inversions or insertion and deletion events can break up this synteny, making it harder to see these conserved pieces of the chromosome. So when you see in bow ties and some future slides, know that that's due to these inversions, right? Okay, but now let's add in some polyploidy on top of this. Okay, let's say one of those species suddenly has a whole genome duplication event, and now there's 2 of everything. Each copy of that genome is called a subgenome. There we're using subgenome 1 and 2. And each one of those genes arising from that duplication event, right? They're pairs from that same ortholog. Those are called homeologs. And when now we do these alignments with this now polyploid species back to the species that's remained diploid, we should see a 2-to-1 ratio of synteny, where for each part of that reference diploid, we see 2 pieces matching in that polyploid genome. Okay. Now, I've been showing you a lot of cartoons, so let's start adding in some real data on top of that. So I study the Tripsacinae, which is this subtribe of grasses in the Anthropogoniae. It includes 2 genera, that is Triticum, which is a wild grass that grows kind of more in the United States and in more northern regions. And then we have the genus Zea, which is mostly centered in Mexico and Central America. And that's made up of a number of wild species, mostly annual, as well as our domesticated maize. And since maize is a very important cultural and economic crop, we have a lot of genomic resources, including 26 genome assemblies that are chromosome level and cover a wide range of diversity, including tropical and temperate material, sweet corns, popcorns, field corns. It's a good encompassing of maize diversity. Okay. And we know that it had a whole genome duplication event about 5 to 12 million years ago. And we know this because sorghum, which has remained diploid over that time, we see a 2-to-1 relationship when we align these genomes to sorghum. So I know this is a complicated figure. We're going to walk through it. So along that x-axis, we have our sorghum chromosomes lined up 1 through 10. And on that y-axis, we have the maize chromosomes lined up also 1 through 10. And each of those circles are representing areas of synteny that had been identified. And so when we look at sorghum chromosome 1, we see that there's a full copy of it essentially in the maize chromosome 1. We also see a second copy of that sorghum chromosome 1 split up across the maize chromosome 5 and the maize chromosome 9. And so we see this expected 2-to-1 relationship that tells us that this was an ancient polyploid at some point. Okay. So because we have all of these many chromosome level assemblies for maize, we've been able to study what's happened to these subgenomes over time. And so, for instance, we know that one of those subgenomes, the maize-1 subgenome, has been preferentially retained, while the other one has been preferentially lost. And this is likely due to functionality differences. Genes on the M1 subgenome are more strongly expressed, right, and are likely contributing more to traits than the M2 genome. We also know that fractionation is ongoing, meaning that if you were to take 2 maize lines and you look at a particular pair of homologs or genes, you might see in one line that both are retained, or one or the other is retained, or both copies are deleted. But you might see a different outcome in that other maize line, suggesting that fractionation is not something that happened just long ago, but is happening also today. Okay. And I'm telling you about all of this because there was this recent project called the Pan-Androbotaniae project that did a lot of genome assembly, not just in this subtribe, but across the entire tribe of the Androbotaniae, which is kind of warm season grasses. And among these genomes that were assembled included a Andropogon virginicus, which is a sister species to sorghum. It's wild, but it has also remained diploid during this time frame, and so can be used as a reference outgroup along with sorghum. We also have 2 new Triticum genomes that are chromosome level, as well as multiple wild Zea genomes. So we can combine all of these wild genomes with our maize genomes and thus get an unprecedented number of assemblies representing descendants from the same whole genome duplication event. I want to reiterate this point because all of the information that we have kind of come to conclude about polyploids, we have done through studying polyploids from different whole genome duplication events and comparing across them. Whereas this one is the same event, but all of these descendants have experienced just this one. So this is really, really exciting because it allows us to ask 2 big questions. 1, how repeatable is fractionation across all of these lineages, right? It's the exact same event. How similar are their responses to it? And 2, we can start to ask questions about when does fractionation really occur relative to these species diverging from each other? And so we're going to focus on this first question first. So I told you that these are chromosome level assemblies, right? And they're some of the first for Triticum. And so we're curious about how conserved that synteny is between sorghum and Triticum. And so important to understand, Triticum has 18 chromosomes. And that ancestral polyploid probably had 20. So there's not been a huge reduction in chromosome number, only losing 2 chromosomes. And so when we compare it to sorghum, each of these ribbons are colored to each of the 10 sorghum chromosomes. And each ribbon represents synteny between sorghum and the Triticum genome. And hopefully what you can see is that for each sorghum genome, chromosome, I should say, excuse me, you see 2 Triticum chromosomes, right? Only a couple where those chromosomes have been mashed together, right, to have that reduction to 18 chromosomes. Okay, so we see this massive conservation of chromosome structure between Triticum and its ancestral polyploid. What about Zea? How is that different, right? So when we look at Zea, right, we're looking at 10 chromosomes, which is very different from Triticum's. So there's been this massive reduction in chromosome number. And the way we get that is because when we compare to sorghum, each maize chromosome is basically a mosaic of many sorghum chromosomes that have been mashed together, right? And so we see these huge chromosomal rearrangements relative to sorghum. Okay. But you might be asking yourself, Sam, you said you had over 30 genomes. You're only showing me 2. What about all those other ones? How does synteny to sorghum compare within and across these genera? So at the bottom are our 2 reference genomes, sorghum and Anthropogon virginicus, and they're strongly syntenic to each other. And likewise, our 2 Triticum genomes are strongly syntenic to each other, as they are to both of our reference outgroups. And at the top is that Maize reference genome B73. So I'm going to start adding in some genomes. Let's start with our wild Zea species, Diploperennis and nicaraguensis. We see that they show lots of chromosomal rearrangements relative to sorghum, but are strongly syntenic to each other. And as we add in the wild relatives that are subspecies of Zea maize, such as Parviglumis, mexicana, and Weiwei tenencensis, we can see that there are some inversions, which are those bow ties, but we still see that synteny is strongly conserved across Zea. And we can add in the rest of our domesticated maize genomes, and hopefully you can see it's a pretty consistent band of color across all of these genomes, indicating that we have really strong conservation of synteny within genera, but very different between genera. Okay. Now, I also told you that this was part of a larger data set, and I would be remiss if I didn't talk a little bit about that. And so this larger data set is looking at the whole Anthropogoniae, and Michelle Stitzer, who's here in the front row, has been looking at what kind of has happened tribe-wide. And so we have been focusing on this one whole genome duplication event, but when we look across the Anthropogoniae, there have been many other polyploidy events. And here I'm just highlighting in red the whole genome duplication events. And when we look at the synteny to their, sorry guys, what you should be seeing is there is huge ribbons of color coming down between each of these chromosomes. Sorry, there's been some file formatting struggles. But what you should see is this kind of very conserved chromosomal structure across all of these genomes, indicating that they look a lot like what we see in Triticum. Another way of saying that is that Zea is an outlier, at least in an observational sense, if not a statistical sense. And I know this isn't particularly convincing, but of that point. but you can absolutely convince yourself by looking at that QR code, which is the preprint for the Pananthropogoniae paper. So please check out that preprint and believe me when I tell you that that's what it shows. Okay, so let's focus back down into our whole genome duplication event in Zea. And here we're really focusing on these potential outcomes for pairs of genes, right? Either they are completely retained as duplicates within a genome, or one or the other have been lost, or both are lost. And we identify these fractionation events by looking for deletions within the exons of genes. So the first thing we have to do is align our genomes to sorghum to identify where deletions have happened in our Zea and Triticum genomes versus sorghum. We also know that one of those homologs should be in the M1 subgenome and the other should be in the M2 subgenome. And we can figure out which is which using orthology and synteny. We then need to combine this information so we know which homologs had which deletions across each genome. Basically, making sure that we're comparing blues to blues and reds to reds across all of these genomes. And we're looking at about 12,000 genes because while all our Zea and Triticum genomes have been undergoing fractionation, sorghum's been evolving too, right? It's not necessarily exactly what it was, 12 million years ago. And so we use that other reference outgroup, Iandropogon virginicus, to come up with these gene models that have the same number of exons across both of our reference genomes, and those exons have the same length, basically saying we think that these gene models are probably highly conserved and are more likely to be representative of what we would have seen in that polyploid 12 million years ago. And so these represent about 69,000 exons. And so just across genomes, when we're thinking about how much of M1 and M2 have been retained versus lost, we see that M1 is preferentially retained, almost 60% to 40%. And M2 is preferentially lost in about the same, kind of the reverse proportions. Suggesting that M1 has been preferentially retained across all of these genomes. And that's exactly what we see when we now look at pairs of exons across each of these genomes, where if one of those exons is lost in a genome, more often than not, it's the M1 that sticks around, and it's the M2 that's lost. But you might also be looking at that section of where we see pairs of exons retained, that both retained. And you might notice that our pink dots, which represent our Triticum genomes, have a much higher percentage of exons observed in that both retained category. This suggests that Triticum has retained more pairs of exons and thus has had less fractionation or loss of DNA than Zea. Okay. We've been talking about exons, but a lot of people, they really want to know what's happening at the gene level. And so we can think about, on average, how many exons from a homolog have been lost. And we can break this down to percentages because not all genes have the same number of exons, right? Keep it standard. And so here in black, we have this reference gene model for 3 exons. And we can think about if 1, 2, or all 3 have been lost, right? You would see 33, 66, and 100%. And so across each of these genomes, what we find in both M1 and M2, the majority of these homologs have been completely lost. There's no exons left. There is a bump where only maybe a little bit of the homolog has been deleted in M1, but for the most part, we see that the entire gene has been deleted. Which brings us to this perhaps philosophical question of, at what point do we consider a gene to be fractionated? How much do you have to lose before you're no longer a gene, right? And I don't have a satisfactory answer for you to that particular question, but I did try some different cutoffs, and I'm going to show you the most extreme, right? And so at the most liberal cutoff, we consider a gene to be fractionated if even a single exon in that gene has been fractionated. Even if all the other ones are retained, if only one is lost, then the entire gene is considered lost. And as you would expect, under this very liberal definition, the majority of our exons are considered lost, but we do see a handful of genes that are completely retained, right? Meaning they still have all of their exons available to them. And then the kind of opposite extreme, where every exon in that gene has to be lost for that gene to be considered fractionated. We see, right, there is this bump in terms of the number of genes that are considered retained under this very strict definition. But we still see a strong amount of genes that are completely lost, meaning there's no exons left of them in these genomes. So kind of the big thing that you should take away from this particular slide about genes is that whole gene loss is a heck of a lot more common than whole gene retention. And this got me wondering, if an exon or a gene starts off on one particular ancestral chromosome instead of another chromosome, how often is the outcome of that fractionation different, right? So are you more likely to be retained if you're, say, on chromosome 1 than if you're on a different original chromosome from here, sorghum? And so what I'm showing you here is for exons, and we take the percentage of exons found on that reference chromosome in sorghum, and we say how many of those exons are considered both retained across our genomes. And what you can see is that if you're on sorghum chromosome 1, you are actually much more likely to be observed as being both retained than any other chromosome, particularly sorghum chromosome 5. And so there's actually a difference in terms of how often genes are fractionated based off of, it seems, which chromosome they originate from. And so we can see that this is true across many other of our fractionation categories. And so there's this large variation between ancestral chromosomes, which brings us to the big question, why? Why do we see a difference? And again, I don't have a particularly satisfactory answer to that, but I do have a hypothesis. And my hypothesis has to do with what happened to those ancestral chromosomes, right? So when we're looking at the synteny of sorghum chromosome 1 versus sorghum chromosome 5, we can see that across both subgenomes, chromosome 1 in sorghum has been retained in pretty large chunks. It's not very broken up, even in Zea, where there's been a lot of rearrangements. But chromosome 5 from sorghum is really sliced and diced. It's in really small little bits and pieces. When you make those dot plots for synteny, it looks like confetti. And they're so broken. And so I think there might be this connection between rearrangement histories of these ancestral chromosomes and the amount of fractionation that we observe today. Okay, so to summarize our answers to this first question, what we find is that all lineages do show this biased retention of the M1 subgenome, and that fractionation tends to affect all of the exons within a gene. And that Triticum retains many more homolog pairs than Zea, and that this perhaps might be due to different chromosomal rearrangement histories. Okay. So this brings us to our second question of when did these differences start? And when did all of this start to happen? And we can look at this in a number of ways. The first way I want to talk about is parsimony, which is this idea that the simplest solution is likely the correct one. So if we were to take our observed states of retained versus lost, if you could only put that fractionation event in one spot on the tree, where would the spot be that gives us this observed pattern? So in the blue case, we only see fractionation of an exon in a single species. And so it must have happened much more recently after that species diverged from the others. Whereas with the red copy, we see that that fractionation event is found in 2 of our genomes, and so must have happened earlier, right? Before those species diverged from each other. That's all we're doing here. And so we're only using exons here that have a definitive fractionation or retention call across every genome. So we're not using the full 69,000 exons for this analysis. But those that we do use, about 80% across both M1 and M2, are completely retained or completely lost across every genome. This suggests that the majority of fractionation must have happened earlier, before the divergence of Triticum and Zea, about a little over 500,000 years ago. And about 10% of those exons can't be mapped to a single point on the tree, meaning that you'd have to put down multiple fractionation events to get to that observed pattern. And so we're grouping all of those instances under this term paraphily. Okay, but if you're doing your math, you might be asking me, what's about the other 10%? And so as we look at those exons that are fractionated across all of Zea, but retained in both of our Triticum genomes, we see that about 4% of exons fit that description. Whereas the reverse, exons that have been lost across both of our Triticum genomes and retained across all of the Zea genomes, is only about 1%. This is more evidence that Triticum has experienced less fractionation than Zea, and that this is likely due to increased fractionation in Zea after they start to diverge from each other, so after that speciations event. But that leaves us with the last 5% of exons. And that is divided into smaller and smaller increments where the exon is fractionated in either a handful of lines or even just a single individual genome. And while these percentages are low, it does mean that recent fractionation was detected across all lineages, and thus fractionation at these exons could be different across genomes. Which gives us to this question of how do you even measure that? And gets us to this term of differential fractionation, which is just a way to capture how different is fractionation for a given locus across these genomes. So when things aren't different at all, where every single genome has the exact same status for an exon, we can say it has one status, right? No differential fractionation. But perhaps, maybe when we look across all genomes, we see 2 of these potential statuses. And that is a little bit of differential fractionation. And we can add and add until we see all 4 statuses across our genomes would be the most differential fractionation that you could possibly observe. And so this is what I mean by 1, 2, 3, and 4 statuses. And so here we have those statuses going from no differential fractionation there at 1 to the most differential fractionation that you could observe at 4. And each black point is the percentage of exons that we see for that particular number of statuses across all of our genomes. And so the majority of our exons, right, only show 1 fractionation pattern, indicating that it's all shared, right, which we had just seen in our previous analyses. But about 35% of these exon pairs show some differential fractionation across genomes, right? Which is pretty stunning. We weren't expecting that at all. We're expecting things to be very similar across these genomes. But again, we've been talking about exons, and everybody wants to know what's happening at the gene level. So here, I have an example of some real data. We have this sorghum gene that has 13 exons, and I've made a representative of the M1 homolog across a few of our genomes, where you can see perhaps in the Triticum, we see many of these exons are retained, but exon 1 and 10 are lost. And when we look across our Zia, we see a lot more exons are gone, and just a few are retained, if any at all. And so as you're looking at this, you might be wondering, how do you even begin to summarize comparisons across the sharing and differences of fractionation and retention across all of these different genomes? Remember, we've got about 30 plus genomes and 12,000 genes to evaluate. And so I've come up with this particular system where you can have these 3 levels where fractionation has occurred in 2 genomes that you're comparing. If that fractionation is completely shared, right, the exact same exons are lost across both genomes, we call that complete sharing. Whereas if only some of those exons have been fractionated in both genomes, it's just some. And if some exons are fractionated in each genome, but they're completely different exons that are lost, right? There's no overlap in which genes or which exons are lost. That's no sharing. We can then also add in the layer of how closely related are the 2 genomes that we're comparing, right? So if they're the most closely related, they're accessions of the same species, right? And on the other end of the spectrum, we would be comparing a genome from Triticum versus a genome in Zia. And in the middle, we'd have some comparison of genomes that aren't the same species, but are not, but are in the same genus, right? So these are comparisons within our Zia subspecies and species. Okay, I go through this to help us understand this particular box plot, because there's a lot of information here. And so when we are looking at homeologs where there's fractionation occurring in both, but that fractionation is completely shared, in the light pink and light green at the top here, we see that there's the greatest amount of sharing. And these represent comparisons between genomes that are closely related, accessions of the same species. And when we look at kind of more diverged genomes, right, comparisons between a Triticum and a Zia genome, we see less complete sharing. And as we look at more differential sharing of fractionation, or less sharing, we see that those more diverged genomes, so our Triticum versus Zia genomes, are much more prevalent in these categories where there's less and less sharing. Also note that it's very, very rare that none of the fractionation is shared at all, right? Most often when we see fractionation between these 2 genomes that we're comparing, there's at least some overlap in which exons are lost. Okay. Whenever we see patterns that are either shared or different across lineages, we want to know why. Why is there sharing and why might there be differences? And so this naturally comes to the question of selection. And DNDS is a way of measuring selection. So on this scale, we have DNDS of 0 refers to purifying selection, which is the removal of bad alleles from a population. And at 1 or much greater than 1 DNDS, that would be positive selection. So promotion or keeping around really good alleles in a population. Now, if differences or changes between 2 genomes, right, if those have a DNDS of 1, that means that those changes are neutral, right? They're not under selection. They might not have a functional or a fitness effect, right? And so any changes that we see there is likely due to random changes, right? Random drift. And so when we look at those genes that show complete sharing between a focal genome and any other genome, we see that the majority of the DNDS values that we have for those genes are at centered on 0, right? Meaning that they're under purifying selection. So selection is acting to remove deleterious variants. But as we look at genes where they have less and less sharing between genomes, we see that that distribution moves further away from 0 and closer to 1, meaning that they are more likely under weakened or relaxed purifying selection, or are becoming more neutral and thus affected by more random changes. Okay, so what does all of this even mean in terms of when fractionation occurs, right? So big picture, the majority of fractionation is shared, right, across all of these genomes, suggesting that it happened before these species began to diverge, so long, long ago. However, we do see a large proportion of exons that show differential fractionation, meaning that fractionation is indeed ongoing and at larger scales than perhaps we expected. And that sharing of fractionation on a gene-by-gene basis seems to be correlated with relatedness, with more related genomes sharing more of the fractionation patterns, and that any differences that we see are likely being driven by relaxed purifying selection. Okay, now I know that was a lot, and so some big takeaways from this study is that fractionation is much more dynamic as a process than we really expected to see. While the majority of fractionation was just shared across all of these genomes, including the biased retention of M1 over M2 homologs, and the loss of entire homologs, we do see these strong differences between members of the Triticum genus and members of the Zea genus, which we did not expect at all. I can't emphasize that enough. We really expected that all of these lineages that descended from the same whole genome duplication event would be very similar to each other. And so we hypothesized that these strong differences might be related to different chromosomal rearrangement histories between these two. And finally, differences in these fractionation between genomes are probably likely driven by drift, right? And are correlated with relatedness. And it's usually at about this point in any talk I give on this subject that somebody asks this question: what about sub and neo-functionalization? And you might be saying, Sam, those are some really big new words. Did you use those in your talk? And I did not. Because we really focused on retention versus fractionation as the potential outcomes of this downsizing. But actually, there are many outcomes for duplicate genes after a polyploidy event. They can be subfunctionalized, which is where that original function of that gene is split across these two copies, either by each copy taking part of the original job, or by becoming tissue specific. So one gene is used for that job in specific tissues, and the other gene is used in different tissues. They could also become neo-functionalized, which is where one copy retains that original function, and the other copy is allowed to mutate and generate new sequence and basically create a new novel function. And these are really cool. They're also really, really difficult to prove and require a lot of molecular work to validate them. And so I chose not to investigate them in this study. That's not to say that these aren't necessarily happening in these genomes, just to say that they would be really hard to study. And I do have a really great example of neo-functionalization where they did do that kind of validating work. So if you're excited about that, this was an instance of sodium ion channels that were duplicated after a whole genome duplication event in fish. And one of those copies of that sodium ion channel neo-functionalized to basically become an electric sensory organ in fish. So if you're interested in that, I highly recommend reading that paper. It's really cool. Okay, but it's not just maize, right? We're plant biologists. We like to think about all of the other many diverse species available to us. And so what we can take away from this beyond just what it means for maize and its wild relatives is that polyploidy and fractionation have shaped every plant genome through these multiple rounds of expansion and downsizing. And so if fractionation is much more dynamic between descendants of the same event than we previously expected, what does that mean for all of our other polyploidy events? and all of the descendants from those. How do we see that this connection between chromosome stability and fractionation when we look at across all of these other polyploids? What might make chromosomes more stable than others? And how we haven't even touched life history or some of these other kind of biological factors that might influence these processes occurring at the genome level. And so since this is a plant breeding symposium, I wanted to have this one takeaway slide of what does this study add to plant breeding? And so hopefully through this talk, you've gained an appreciation for how fundamental these processes are for generating the diversity and structure of modern plant genomes. And that while many outcomes of these processes are shared across lineages, descendants of the same whole genome duplication event may have very different outcomes in terms of what diversity is kept or lost. And finally, that these processes develop novel, potentially adaptive variation beyond what was available in those parental species. With that, I would like to thank all of my funding agencies, mainly NSF, who have supported this work, all of my wonderful co-authors and lab mates, my PhD committee members, and the Practical Haplotype Graph team for their help and support in completing this work. I'd also be remiss as a postdoc not to mention that I'm working on a completely new project with my advisors, Jeff Rossi-Barra and Graham Coop at UC Davis, looking at human domestication of maize as a biocultural coevolution. I'd be happy to discuss this project more with folks during the social hour later, but I just wanted to mention that that's what I'm working on currently. And so with that, thank you so much for your time and attention. I'd be happy to take any of your questions.
We're over time, but I'll take like one question.
All right, I wanted to ask what your greatest challenge is in this research and what you're most excited or passionate about.
With this research? Yeah, so I think the hardest part about this was developing the methods, right? Trying to come up with a way to accurately identify fractionation and that it be the same fractionation across each of these genomes. That was really challenging to use these tools that might not have been built for that particular purpose, but I use them for it. And what I'm really excited about is just the amazing diversity that we can study given all of these new genomes and genome assemblies. There's so much big data out there. There's so many cool questions that this data allows us to address and answer from basic biology that we previously couldn't really get at. And so I'm really excited about the opportunities that big data give us.
Cool, thank you so much.
Okay. One last round of applause.
So we are approaching the end of the symposium. I would like to thank all the speakers for taking their time to come all over to Ithaca and give these wonderful presentations. I'm really grateful to them, and also for the audience. I'm grateful to you for giving your presence here and making this symposium a success. Lastly, I would also like to thank my planning committee members who have been a source of support to the symposium and to me. With this, I would like to say that we have a social hour outside, and with this, we can end the symposium. Thank you.
This has been a production of Cornell University, on the web at cornell.edu.